Hello, everyone. Today we're going to circle back to major body organ systems, but we're going to zoom in or focus on the versions of these systems that vertebrates have and principally that humans have. We will continue to look at a little bit of the diversity among vertebrates to help us understand how evolution has shaped these human organ systems. And there's a lot to cover in this slideshow, so you might want to take this lecture in a couple parts. We've got 4 major organ systems: digestion, respiration, circulation, and osmoregulation, excretion. So this slide is a reminder that these major body organ systems in a large terrestrial animal in particular each have a specialized job, and that they have to work together in order to meet the needs of the organism. 3 of these systems, the digestive system, respiratory system, and excretory system, are connected to the outside world. They have a job of taking things in from the environment and or putting things out into the environment. So as we're thinking about the basic function of these systems, what comes into the body and what goes out of the body is a good way to start. Now, the 4th system, the circulatory system, is going to stay inside the body. It's not good if we get a leak in it. We don't want it connected to the outside world. Its job is to move substances around and help all of the different body organ systems share the resources that they're getting with all the other body organs, and also to transport communication molecules around. And so this circulatory system, especially a closed circulatory system where the body fluid, the blood, stays inside the blood vessels, its function is transport. Right, moving things around. The respiratory system is going to focus on gas exchange in heterotrophic animals. That is going to be oxygen in and carbon dioxide out. We're going to have the digestive system, which is going to take in food. It's going to process and digest that food and then absorb it. So molecules have to move from the inside of that tube that makes up the gut into the body cells and into the transport system. So those blue arrows that are showing things moving into and out of, we have to have this movement from a body system into the circulatory system and also out of the circulatory system. Now, the excretory system is going to be taking metabolic waste products. So remember, metabolism is a way of saying all the chemical reactions that happen in the body. So chemical reactions that occur during the processes of digestion and breaking down and building other molecules inside the body are going to generate some kinds of waste products. So molecules that are products of chemical reactions that are either excess or extra, things we don't need in order to maintain homeostasis. We want to get them out of the body. Other materials that are toxic or harmful, we want to get rid of those as well. So our excretory system, the excretion part of it, is the getting rid of or moving outside the body. The osmoregulatory part isn't shown here because it isn't principally concerned with moving things into and out of. It's concerned with managing homeostasis of water. All right, so things moving out of the digestive system are undigested, undigestible, and or unabsorbed matter, and we call that matter fecal matter or feces or poop. So it's going to consist of, in humans, it's going to consist of a lot of plant cell walls. It actually consists of a lot of your own body cells and bacteria. as well from the populations that live inside of our gut, almost up to half, in fact, which is mind-boggling to me. All right, let's look at each of these systems in more detail, starting with the digestive system. All right, we have 4 stages of food processing: ingestion, taking into the body, stuffing food into our mouths. We have digestion, which is breaking down the food so that it can be absorbed. Digestion has 2 components. One is mechanical, and in mammals, this is done by chewing. So with our teeth, we're going to take bites and we're going to chew the food. We go from having large pieces of food to having smaller pieces of food that go down our digestive tract once we swallow. After mechanical digestion happens, then chemical digestion is going to be able to proceed. Chemical digestion is via enzymes breaking chemical bonds, right? So mechanical digestion is physical, chemical digestion is chemical. So that's at the molecule level. As we think about how food is processed, we're going to be eating things that are organic, that are other plants or animals, and that their bodies are made up of the 4 kinds of biological macromolecules that all life is made of. So remember that we have these 4 kinds of macromolecules. We have lipids, we have proteins, we have carbohydrates, and we have nucleic acids. When we think about what enzymes do, remember that each enzyme has one job that it's able to do. It has specific substrate that it is going to attach to and be able to do one chemical reaction. So as we're looking at different components of our food mixture, we have to have a different digestive enzyme that is able to break the chemical bonds between the different biological macromolecule subunits. For example, when we have a protein, it's made up of a long string of amino acids. So we need an enzyme that can break amino acids apart from one another. When we have carbohydrates, they're going to be a long chain of monosaccharides or single sugar molecules bonded together. We have to have enzymes that break apart those single sugar molecules. We need a different enzyme if we have 2 glucoses bonded together than if we have 2 fructoses bonded together. Okay, so we have lots and lots of different digestive enzymes because we have many different types of molecules to digest. All right, we want to break those big pieces of food down into small pieces of food so that as the enzymes are starting to attack them, there's going to be more surface area. So if we swallow a large object, or when we look at other organisms that have no ability to do mechanical digestion, the digestive enzymes can only start to pick at the outside of that big blob. Where we break apart that big blob into many little blobs, then we have increased the surface area that can be attacked by those enzymes. All right, so more efficient digestion means that we can get more nutrient molecules out of the same amount of food. Taking in food requires searching for it, finding it, perhaps fighting with it. So the more efficient we can be with getting all of the resources out of the food that we did get will help us save energy and be more successful. All right, so absorption is the next step. So think about the fact that the gut is a tube. It has epithelial cells lining the outside of it, but also lining the inside of it. So when we say absorption, we mean that a nutrient molecule that's inside the gut has to go across a cell membrane of an epithelial cell. And in a large organism like ourselves, it then has to go from the back of that epithelium into the circulatory system. So we'll see that there are capillaries or small blood vessels associated with our the regions of our gut that do absorption, so that we can pass those molecules into the main transport system in the body right away. All right, so if material does not get absorbed for whatever reason, it will continue to pass through the digestive tract and it will get eliminated. All right, we got to have a way to get that extra stuff out. Right, there's going to be leftover material that we can't digest, can't absorb. You have to get it out and make room for new matter to come through the system. Right, that step is called elimination. Sometimes it's also called excretion. And that we have the process of digestion and absorption has to meet in heterotrophs 3 different needs. We have to get all of the energy that we need. This is going to be primarily in the form of carbohydrates and fats. We have to get all the molecules that we're going to turn into molecules that we need in our own body. So when we think about the way that biological macromolecules are built up out of smaller subcomponents, right? So the subcomponent of proteins is amino acids. We break proteins that we eat down into separate amino acids. Amino acids get absorbed, transported throughout our body, and wherever we need to make a protein, right, which is going to be every single cell in the body, because every single cell has enzymes in it, and enzymes are made of proteins, right? Those amino acids get distributed, and then they get bonded together by the ribosomes based on the messages from the DNA to make whatever proteins are needed in each cell. So we need to get what are called those molecular building blocks. You can think of them as carbon skeletons. You can think of them as those amino acids, monosaccharides, fatty acids, and nitrogenous bases, things we have to build into whatever we exactly need in our own body. So we need enough of those that we can do all the building that we need to. We also need a certain small amount of a variety of nutrients that we can't build or make in our own bodies. So we have to take them in our food, and they're called essential. There's a couple of amino acids that are essential because we cannot make them in our body. And then we refer to things of vitamins and minerals typically as essential because we cannot make them in our bodies and we get them from our diet. So specifically thinking about humans, I would say that because of our large brains that build meaning about things, and that we act and we live in societies and cultures, we eat for a lot of reasons besides the fact that we need nutrients. We eat because we enjoy it, because it's pleasurable to have food that tastes good, and for a whole long list of social and cultural reasons. We may have foods that help us celebrate religious festivals, or that we have ways that we interact with others in our social groups that revolve around food. So foods can have, just for humans, a wide variety of other purposes. But in order to live, to survive, our bodies need to have energy, building blocks, and essential nutrients. When we don't get the nutrients we need, overall malnutrition is the word that's used for not getting enough nutrients. And there are specific diseases or disorders that come from having missing certain essential nutrients. So here's a couple examples. Now, in developed countries, these are relatively rare because we have all kinds of public health policies that have happened in the past in order to help solve these problems. We also have the availability of fresh fruits and vegetables year-round because of preserving, refrigerating, and freezing, and also transport that moves For example, fruits, we're drinking our orange juice, eating our oranges year round because we get them imported from different areas. So goiter is a swelling of a gland in the throat region that happens if there's insufficient iodine, the thyroid gland. If you look at the salt boxes where you get salt from the grocery store, most of them say they're iodized. So in the US, just eating a diet with food that's been salted is going to provide sufficient iodine because of a public health measure, which basically the government said, hey, salt manufacturers, make sure there's iodine in your salt and we can prevent goiter. If you only use those fancy sea salts, you may be at risk of getting insufficient iodine, right? And this can be a problem in developing countries where people are using local sources of salt, or they don't eat a lot of seafood, which is also where we can get iodine. Rickets occurs in children who have insufficient vitamin D in their when they're growing, and it causes weakness of the skeleton in the legs and bending of the legs as a result. We are capable of making vitamin D if we get exposed to sufficient sunlight. And we also get it in a variety of foods. And in the United States, we have had, again, public policies where we have fortified or added vitamins to different foods that people are likely to eat. So we have it added to milk, we have it added to other products that people are likely to be consuming, especially as children, to try and prevent this. Scurvy is what happens if you have insufficient vitamin C. It's pretty easy to avoid, but it used to be really common among sailors who were at sea for long periods of time and were not eating sufficient fruits and vegetables. It can lead to a variety of breakdown of tissues, and if it's severe enough, it can cause loss of your teeth and even death. Another example that's pretty uncommon in the developed world is beriberi, which is due to insufficient B1 vitamin. All the B vitamins we typically get from whole grains and also other common foods. So if we have a sort of normal American diet with, breakfast cereals, those are also fortified or supplemented with some of these vitamins that we would otherwise have an insufficiency and some kind of disease as a problem. All right, so when we're talking about the human digestive tract, it's basically the same as the digestive tract of all vertebrates. Once we get to those fish-like vertebrates, we have basically the same body organs. So we think about the digestive tract itself. It's a tube, and it grows during gastrulation in the embryo. If we have a complete digestive tract, we have a mouth and an anus. So the mouth opening is going to be the beginning of this tube. The tube goes continuously through the body all the way to the anus, which is opening to the outside world at the other end. We divide the parts of this tube. They have become specialized to have different jobs. We're going to see the digestion happening first, and then the absorption, and then the elimination. And besides having regions of this tube, we also have what are called accessory glands, right? When we think about the fact that we may accessorize our outfit, or we have accessories, those are going to be our purse, our jewelry, our watch, our belt, whatever, things that are hanging off the side of our otherwise outfit. Right, our accessory glands are basically hanging off the side of our digestive tract, and they're going to be like our endocrine glands. They're going to be secreting something, putting something into that digestive tract. All right, so the cells of the digestive tract itself are often secreting materials, but we have some important components that help the digestive process happen. due to these accessory glands, right? The parts of the tube itself start with the mouth, the oral cavity. Behind, or the next step is the pharynx, right? And then the esophagus. These are basically sort of stretchy tubes that connect the mouth to the stomach. The stomach is going to be a sort of a temporary resting place for the food before it gets moved into the small intestine. The small intestine is small in diameter, right? It's very long compared to the large intestine, but it's small in diameter. The large intestine is large in diameter, but shorter. The small intestine is sort of all squiggled up, and it has a couple different parts. The very uppermost part of it is called the duodenum. The large intestine has a couple parts as well. It has this ascending or going up portion. So it joins to the small intestine kind of on your lower right-hand side. That's where your appendix is, that little thing dangling down at the edge of the beginning of your large intestine. If you have sharp pains that are ongoing in that lower right-hand corner of your abdomen, you should go to the emergency room. You could have acute appendicitis, and it's life-threatening. All right, so we've got ascending colon, transverse going across, and descending. And then there's this tricky part that squiggles around and turns a corner here to the rectum, which is called the sigmoid colon. The rectum is a place where compacted fecal matter is going to be temporarily stored before you pass it out of the body. So notice we have a variety of other structures associated here. As we're going through the tube of the gut, or the alimentary canal, there are various places where there's a structure called a sphincter, right? A sphincter is similar to a valve in that it's going to be a little ring of muscle. Now, valves are usually sort of flaps. A sphincter is a ring of circular muscle. When that ring of circular muscle is contracted, it closes up the opening of the tube, right? So instead of having an opening, we have a closed place. And that's important in order to stop the movement of material through the gut. We either don't want it going backwards, or we don't want it going forwards, because we want it to stay in a particular location, right? Besides having sphincters at the top of the stomach, which is called the cardiac sphincter. Sphincter at the bottom of the stomach is called the pyloric sphincter. We also have an anal sphincter, right? That's the muscle that allows you to hold on to your poop, and so that it doesn't just dribble out whenever some is ready to go there, right? That's incredibly convenient, right? We might have a predator that is able to find us because of our leftover products that we have dribbled around. And so that if we can control where we release those, we may be able to avoid predators. We can keep our living spaces clean and sanitary. And we have a whole variety of instinctual behaviors that remove our fecal matter and urine sort of from the places where we go through our normal daily lives, and especially food preparation and sleeping. All right, so sphincters are very good and useful things. Of the accessory glands that are going to be adding things to our mixture of food going down the digestive tract, we start with the salivary glands in the mouth. They make saliva. They make a lot of it, several liters a day. That's going to be added to the material we're chewing in our mouth. It adds liquid in order for us to be able to swallow it and for it to mix and be mixed with the enzymes and other secretions that we need to have access to all of the little food particles. Saliva also contains an enzyme that's going to start digesting carbohydrates. It's called salivary amylase. And an amylase is an enzyme. So all the ASE endings, remember, those are for enzymes. Saliva also contains lysozyme, which is an enzyme that breaks apart or kills bacteria. Remember that as we're taking in food from the environment, we may also potentially be taking in pathogens. So the digestive system has built-in protections from pathogens to help us not get sick from the food that we are eating. All right, so we have in our mouth, we have this lysozyme to help kill bacteria that we may have taken in with our food. All right, we'll go through a little more detail of what happens after as we are swallowing our food in a minute, but I want to go through these different accessory glands while we're on this slide. The liver is the next major accessory gland, and it is an incredibly dynamic and complex body organ. It's a powerhouse of biochemical functions. It has over 500 chemical reactions that it does. Specifically related to digestion, it's going to produce a substance called bile. Bile is going to be produced in the liver, and then it's going to go into the gallbladder. So the gallbladder, like our urinary bladder, is a place that temporarily stores things. It temporarily stores this material called bile, which is some fluids and some bile salts. Those are going to get added into the mixture of material that's in your small intestine. And the function of these bile salts is going to be to help make fats in our diet get dissolved into the watery solution that's in our gut to be able to make them, like we can chew fat, but we can't make it dissolve, right? Because it's not water soluble. So the bile salts will help make those fatty food particles mix in with the water so they can be further digested. The liver also is going to mix some other stuff into the contents of the intestine, and that is in order to get that stuff eliminated from the body. The liver breaks down old red blood cells and disposes of them. And remember that the red blood cells are red because they have a molecule that's inside them, hemoglobin. Hemoglobin is called a respiratory pigment molecule. It's called a pigment because it has a color, or it looks a color. That color is red. And as we break down those hemoglobin, there are these pigment molecules that are left over and broken up and disposed of in the gut. So they actually predominantly determine the color of our fecal matter. The liver also helps to detoxify and remove molecules that we have absorbed from our gut. So anything we eat as food that's potentially toxic, it's going to go to the liver, and through a series of chemical reactions, it will get transformed into a substance that's less toxic. Can get stored in the liver, but it can also get eliminated. We can dump it out into the small intestine, basically via the gallbladder. All right, so lots of neat stuff the liver is up to. You'll have to wait for another class to talk more about it. All right, the pancreas is really important for a couple of reasons. One, that we already talked about, it secretes a buffer, that is a solution that's going to normalize the pH. The material coming from the stomach is acidic. The stomach has protections from that acidity, but the small intestine does not. So as we are moving that liquidy stuff from the stomach into the small intestine, the pancreas is going to squirt in a mixture that includes bicarbonate ion, HCO3-, which acts as a buffer. It's going to absorb the hydrogen ions that are in that acidic mixture and return the pH to close to neutral. The pancreas also contributes a wide array of digestive enzymes. So we'll see in a couple minutes that we start digesting carbohydrates in the mouth with that salivary amylase. We start digesting proteins in the stomach with a digestive enzyme called pepsin. By the time all that material moves into the small intestine, we are done with the chemical digestion. We need to keep doing it. And the pancreas is going to secrete enzymes that are going to break down all the different kinds of molecules that could occur in our food. So it's going to secrete pancreatic amylase to continue digesting the carbohydrates. It's going to secrete an enzyme that breaks down proteins called trypsin and other protein digesting enzymes. And it's going to secrete lipases, enzymes that break down fats, and nucleases, enzymes that break down nucleic acids. All right, so let's talk a little more about what happens as we take in food, how it gets into the stomach, and what the stomach does. All right, when we have food in our mouth and we swallow it, that is a mechanism that's under our conscious control. But once we have swallowed, so we're going to see this green blob here is representing the food, we've chewed it. And once we swallow it, it becomes sort of a blob of food that we're going to call a bolus. It's going to move down our pharynx and esophagus through actions of our autonomous nervous system, controlling those smooth muscles that are around the esophagus. They're going to alternately contract and relax in such a way that it massages or moves this bolus of food down into the stomach. That same contraction of longitudinal and circular muscles that earthworms use for locomotion, called peristalsis, is going to be used by our smooth muscles surrounding our gut, in fact, all the way down through the large intestine to help keep mixing and pushing along through the process those food particles. Right, so we have an arrangement of tubing in our throat where we have the ability to breathe in through our nose or our mouth and have air go down into our lungs. That's quite handy when we have somebody's, we're holding our nose or it's really stuffed up, we can breathe through our mouth to back up. However, it does mean that it's possible we could take in some food and it would go down our airway instead of our esophagus. So we do have a structure that's a flap or a trapdoor that comes down and it covers the opening to the trachea. Right? The trachea is the tube that goes to our lungs. So we see here when we are not swallowing that this structure called the epiglottis, epi means on top of, right? What is it on top of? The glottis, all right? The glottis is the opening, the top of the trachea. When we swallow, there's an automatic response to pop the epiglottis down, and that causes the bolus of food that we've swallowed to bypass the top of the trachea and save us from choking. All right, this isn't a perfect. Anybody who's never choked in their life, raise their hand. Yeah, I didn't think so. Right, occasionally we do end up getting food in our airway. And so this mechanism is imperfect. And remember that we evolved from those early fish-like chordate ancestors where when we look down their mouth, we see the gills going to the side and we see the tube going down into their gut. So we kind of have these two systems for respiration and for feeding that are evolved in our bodies that were right next to one another. All right, so we don't want to try and breathe and swallow at the same time. That's an important safety precaution to avoid choking. All right, so when we get the food down to the top of the stomach has a valve at the top of it. And as food is pushed by the contraction of these circular muscles, it'll squeeze it through that sphincter. Sorry, it's a sphincter into the stomach, right? Normally, after we have eaten, this sphincter is contracted, right? That allows us to, if we wanted to, right after we've eaten or drank some water, stand on our head, right? Do something upside down, and we don't have the food just flowing up back out of the stomach. Important safety precaution for a variety of reasons. Once the food gets into our stomach, it's mixed with this very low pH solution. It becomes acidic. The stomach lining is resistant to that acidity, but the lining, the cells of the esophagus are not. So they would become damaged by that material. Now, occasionally, we need to vomit. So why would we want to do that? If our cells in our stomach detect some bacterial toxin that came in with the food, then the most efficient way to avoid getting infected further by that bacteria is to vomit up the food that we ate. So if we've eaten bad food, we feel nauseated, we vomit. Not pleasant, not something we enjoy doing, not good in the long term. It can cause that burning sensation, which is the acidic contents of the stomach, causing damage to the tissues of the esophagus and the mouth. So we don't want to do it often, but it's an important safety mechanism to help get rid of potential pathogens. So if we have some kind of dysfunction of that sphincter that's congenital, or we have a habit of eating too much food, or too much greasy foods, some kinds of slumping after we've eaten, it can cause some persistent backflow of the acidic stomach contents and can cause damage to that sphincter and the lower esophagus. That sensation, that burning sensation of our lower esophagus is often referred to as heartburn. If it's consistent and prolonged, it's referred to as gastroesophageal reflux disease, or GERD, G-E-R-D. One of the most common over-the-counter medications that people take, besides for pain relief, is for this acid controllers. All right, so when babies are born, that sphincter is not yet fully developed. So this idea that, you know, babies are sort of likely to spit up, it's not entirely their fault. They don't have that mechanism fully formed yet. It finishes, you know, as they continue to develop. All right, what happens in the stomach? All right, so once the food gets dumped into the stomach, it's going to be mixed with more mucus and secretions from the stomach. We'll get to those in the next slide. The stomach itself is stretchy, right? So when we see it relaxed, it's going to have these very sort of deep ridges and folds on the inside. That helps allow it to sort of inflate or get bigger in volume when we eat a large meal. Part of the reason we have a stomach is to allow us to take in a large amount of food when it's available, and then rest and digest, so we don't just have to eat continuously. The stomach's job is going to store and mix food, and what it's going to mix the food with is digestive enzymes that are specifically to digest proteins, right? The other thing that happens in the stomach is the secretion of hydrochloric acid. This is going to increase the acidity or decrease the pH. There's a couple of reasons to do that. One is that we're trying to digest proteins. Proteins become denatured, that is, their three-dimensional structure unfolds when they're in a low pH environment. And so helping to denature the proteins allows the protein digesting enzymes to access their structure so that they can snip them apart. The hydrochloric acid also is a way to help suppress the growth of bacteria that may have gotten into our stomach with the food. All right, so this acidic environment, these protein digesting enzymes actually can cause damage to our cells, our own cells. So the stomach has a few mechanisms that are going to help minimize the damage that the digestion of our food is actually causing to the body part itself. All right, so there's a couple things. One is that the interior of the stomach lining is covered with mucus. It isn't shown in this slide, but there's an especially thick mucus that's secreted on the surface of the stomach. The second is there is damage to the stomach lining, and so the cells have a faster rate of turnover and growth by mitosis than other parts of the body. The third mechanism is structural, and that is that the acid and the digestive enzymes are secreted not directly onto the surface of the stomach, but down in these sort of pits or these tubes that go into the stomach lining. So the pepsinogen molecule is secreted. The hydrogen ions and chloride ions are secreted, and they move with the flow of mucus up into the contents of the stomach. As that's happening, they're going to get converted into hydrochloric acid and this pepsin molecule. So what's secreted is not an enzyme that digests proteins. It's a molecule called pepsinogen. It gets converted into pepsin by a process that is catalyzed by the acidic environment. All right, so pepsinogen is a protein that's very similar to pepsin. It's almost pepsin, but it's not active. Right? It gets converted into pepsin when it mixes with this acidic environment, and then the pepsin itself also helps convert or catalyze the transformation of pepsinogen into pepsin. All right, so hopefully by the time this process of transformation has happened, the pepsin has become mixed up with the food in the contents of the stomach rather than smashed right up to the surface of the cells, because the cells membranes themselves have all kinds of proteins embedded in them. We don't want to digest our own cells. All right, as we see a lot of examples in the body, we regulate the presence of some active version of an enzyme or a protein by first secreting a precursor molecule for it, that then under certain circumstances will get converted into the active form. Right? So we have an inactive form, gets converted to the active form. And a lot of these molecules are going to have the name that ends in the gen, like a progenitor or something that comes first. All right, so as we move the material that was in the stomach, which once it gets in the stomach, it's called chyme, which is spelled C-H-Y-M-E. It's acidic, right? It's going to get moved into the small intestine. The pancreas is going to secrete additional enzymes. It's going to secrete that bicarbonate ion. It's going to neutralize the pH. And we're also going to have the bile getting added to the small intestine. So as those food molecules are moving into the small intestine, we're going to see that larger chunks, larger globs of fat are going to get attacked, the surface of them, by these bile salt molecules, which is going to break them down into smaller particles and then even smaller particles, right? So this process of mixing oils with water, basically, is called emulsification, right? Emulsification is the formal word of sort of making something that is hydrophobic, making it mix with water. When we make mayonnaise, we're taking oil, and we're making it mix with watery solution, which is actually, if you make it the French way, it's egg yolks. And we cause those materials to be permanently mixed together or stably. It's not a chemical reaction. It's a stable mixture that isn't going to separate again. If we take salad dressing, we make our own vinaigrette. It's vinegar and it's oil. And if we let it sit there, the oil is going to separate. It's going to sit on the top. You shake it vigorously. What we've done is temporarily emulsified those two materials just by physically making them mix with one another. But that's not stable. It doesn't stay like that. So what we've done in mayonnaise is we have made a stable emulsification. All right, so what's happening in the small intestine? We're going to continue chemical digestion. Most of that is done toward the top of the small intestine. And we're going to start absorption. There's very little that gets absorbed in our stomach. It's like alcohol and aspirin. Those are the only things. Very little. All right, so absorption, that's going to be the movement of substances across the cell membrane of the cells that are lining the small intestine. All right, now the job of the small intestine is absorption. So just like in our respiratory surface, we want to have a very high surface area because we want to be able to get a lot of molecules across those cell membranes. So the more cell membrane we have, the more molecules we can get across. All right, so the small intestine does this in a variety of ways. All right, so packed into your lower abdomen, take a look down there, see how big it is an area the size of a tennis court. And the way that that is possible is that we have a series of structural adaptations that increase the surface area, and they're stacked on top of one another, kind of like a fractal, if you know what that is. All right, so the small intestine is small but mighty, right? Remember, it's small in diameter, but it's long, right? So we just let the food pass through it for a long time. Then it has a physical structure on the inside, which are these folds. These folds are called plicae or pleats, spelled P-L-I-C-A-E, right? And they're basically the same idea as when you take a potato chip and you make it waffly, right? Ruffles have ridges. Why do ruffles taste better? Why do they taste better? Right, so we've taken a potato chip, right, a flat one, and we've fried it in oil and we've sprinkled salt on it. All right, it's the oil and the salt that taste good, right? So imagine instead of a flat potato chip, we've taken a potato chip that was twice as long and we smooshed it up so that it has these little wiggles across it. All right, so we look down on the top of it and it has the same circumference, but we've doubled the surface area. All right, so ruffles are so delicious because they have twice the amount of fat and salt packed into the same area, right? So same thing with our ridges on the inside of our small intestine, we've increased the surface area. All right, we don't stop there though, right? Now, if we cut open the small intestine of a human, you can see those folds with the naked eye. If you put a dissecting scope up to it and you look at the surface, you're going to see the surface has basically what looks like all these little sort of nubs or tips. It turns out that those are finger-like projections, right, sticking up from the surface. They're not visible to the naked eye, right? So they're multi-celled, however, right? So those are structures that they're each going to have a little capillary going through them. We'll see how that looks in the next slide. So these little projections or little things that stick up are called villi. A single one is called a villus. Right, so we've got these little villi sticking up. All right, now behind the wall of the small intestine, you see that there's a circulatory structure here. So blood vessels are being shown, veins and arteries. or capillaries, right? So small veins and arteries. And when we look at how the circulatory system, it's going to traverse the entire small intestine. It's going to have smaller blood vessels that wrap around it so that it can absorb those nutrient molecules directly from the intestine. However, most of the blood flow going from the intestine isn't going to go straight to all parts of your body, right? It's going to go straight to your liver. Remember that I said the liver has a job of doing all kinds of chemical reactions. Any kind of molecule that we ate in our food that's potentially toxic or harmful is going to go straight to the liver, and the liver is going to be able to identify it, grab a hold of it, do its chemical reactions onto those molecules so that they become detoxified, right? It's also going to manage the glucose and other carbohydrates and the fats that you are absorbing from your gut. So the liver is going to store and process those nutrient and energy molecules to help regulate our blood sugar. All right, so this hepatic portal vein, anything that has to do with the liver is called, often called hepatic. All right, so that goes straight to the liver. The other thing that your small intestine is busily doing is secreting enzymes, and especially in the upper end of the small intestine where the chemical digestion is going to proceed, absorption is going to happen more and more towards the lower end of the small intestine. And so the cells of the small intestine themselves are going to secrete more digestive enzymes. And primarily it's going to focus on breaking down smaller component parts of those biological macromolecules. So by the time the food is getting into the small intestine, we don't have proteins that are thousands of amino acids long. We may have dimers, right? So we may have dipeptides. So there's just two amino acids fastened together. We have a special enzyme that snips apart those last two amino acids from one another. Same thing with our carbohydrates. We may have disaccharides, or molecules that are made up of two of those carbohydrate monosaccharides. So we have special enzymes that snip them apart from one another so that they become monomers. There are also enzymes that are going to do the same thing to nucleotides to break them apart into those nitrogenous bases, phosphate and ribose, and also with lipids, right? However, we also have on the surface of the cell membranes of those small intestine cells, we're going to have proteins, right, that are going to be used to transport those molecules from the lumen, or the space inside the gut, into the cells, right? So this is the absorption step. We've got to take a glucose, which is still a pretty large molecule, and we've got to put it inside the cell, right? So these are transport proteins, and they require ATPs in order to do that work, right? So absorbing all those food molecules is active work. The cells are going to use a lot of ATP in order to move those from one side to the other. Some of those transport proteins also have a job of breaking some bonds, all right? So they're a combo transport protein and enzyme that is going to, while transporting from one side of the membrane to the other, it's going to break a chemical bond, all right? So we have a wide variety of different ways of finishing the chemical digestion and doing the transport. All right, so now we're going to zoom in on one of these villi and see what its structure looks like. All right, so here we have a villus. All right, so this layer of cells on the outside is the epithelium. Coming up through the villus is part of the normal circulatory system. So a little capillary bed is going to wrap around sort of the inside of that villus so we can pass nutrient molecules through the back of the epithelium and put them into the capillary system. These capillaries are also delivering oxygen to the cells lining the small intestine, because all cells everywhere in the body, including those lining the small intestine, need to have oxygen delivered and also other waste products taken away. All right. So notice that we have another kind of structure that comes up into the villus as well. These are little tubes called lymph vessels. And specifically, these little dead end tubes that go up into the villi are called lacteals. Right, the lymph system is kind of a shadow or secondary version of a circulatory system that vertebrates have. The lymph system is primarily thought of as a system that is responsible for immune functions, but it also has some important functions in transport, especially of fats. Okay, so the lymph system connects to the circulatory system. So everything that's in the lymph is going to eventually get dumped back into the circulatory system. So they're not separate from one another. One of the other jobs the lymph does is we do get some leakage of this watery material, plasma, from the capillaries. Most of it gets sucked back in to the capillaries. But if there's any extra that's left over and we have some swelling, then the lymph system is actually, the pressure in that tissue is going to force it into the lymph system. The lymph system is going to move it away. So it helps return interstitial fluid to the circulatory system as well. All right, so we have both the circulatory system and the lymph system are ready to collect material that is getting absorbed from the small intestine. Now let's zoom in to one of these epithelial cells. All right, so notice that we're zooming in over here. So each of the epithelial cells has little ridges or little projecting structures on its cell surface. All right, these are called microvilli. Right? Remember, the villus is the finger-like projection we can see with the dissecting microscope. The microvilli are at the level of the cell, right? Microvilli exist on each cell. And basically, if we look across the surface of that cell on the side that it's on the lumen of the small intestine, each cell has these little projections sticking, almost like cilia, except they don't move, right? They've got these little extensions of the cell membrane. When we look at it from the edge, like we do in histological preparations, it looks like the edge of a toothbrush, right? So sometimes this set of these little microvilli is called the brush border, like a toothbrush, right? It's at the edge of this tissue when we see it in a cross section. All right, all kinds of exciting things are happening at that brush border. Right, that is the cell membrane of the cells that is facing the lumen. So that is where the absorption is happening. And so we have all these little extensions. We have all this extra space to have all those transport proteins. An example of something that happens at the brush border is the absorption of fats, right? So one kind of lipids called fats or triglycerides, the way that they are absorbed by the brush border is that the enzymes in the lumen of the small intestine break them down into fatty acids plus a monoglyceride instead of a triglyceride. Those molecules are small enough to be transported across the cell membrane, but for some inexplicable reason, there are enzymes inside the cell that then convert them back into triglycerides. These triglycerides are not going to mix with water, so they're not going to be able to circulate in the capillaries, right? And so what the cell does is that it compiles together a bundle of these triglycerides, including other phospholipids, cholesterol, and proteins, into a ball that's called a chylomicron. And chylomicron is going to have phospholipid heads on the outside. All right, remember, those are the molecules that make up our cell membrane, and they've got the phosphate head that mixes with water. And then they've got the fatty acid tails on the inside. So what they're going to do is they're going to form a little ball. They're going to put the triglycerides and the cholesterols on the inside of it. And then there's a mechanism for these chylomicrons to be moved through the cell membrane, kind of like exocytosis. And the lacteal, remember, that's the little extension of the lymph vessel, has openings in it that are much bigger than the openings in a capillary. And so the chylomicron can move right in there. All right, so chylomicron is a very generic term for some of these little bundle, almost like a vesicle, that is made up of these lipids, and they're going to have proteins associated with them as well. All right, so here's the moment where we're going to talk about what's in circulating, how are fats circulating in your bloodstream. So these go to the liver, and the liver is going to process them into something they're called VLDL. So they change from being sort of a generic chylomicron to being very low density lipoproteins, right? We've got lipids together with proteins, so we call them lipoproteins, LPs. Very low density. That means they don't have very much protein associated with them. Those get transported to fatty tissue for storage. They can also get converted into low density lipoproteins, or if they're transported to other tissues, they can get transformed into high density lipoproteins. The high density lipoproteins actually go around our circulatory system scavenging up cholesterols, right? And so those are considered, from the perspective of our cardiovascular health, they're considered the good cholesterol because they're scavenged or scooped up by these HDL, high density lipoproteins, and eventually returned to the liver for further processing. Right, so these chylomicrons start out as how we get these water insoluble molecules into our circulatory system. It's via the lymph. All right, so we've gotten all our absorption done. We're moving this undigested, undigestible, leftover waste into the large intestine. Right, the function of the large intestine is to help recover water. Now, as we're salivating, as we're putting all these accessory solutions into the gut, and materials moving through it, we've added about 7 liters of water to the amount to the digestive tract. We need to reabsorb it so that we don't get, first of all, that we don't have diarrhea, and second of all, so that we don't have excessive water loss. We reabsorb almost all of it in the large intestine. The way we do that is the large intestine actively pumps sodium ions out of the cells, and that pulls the water after them into those cells, and it gets passed into the circulatory system. When we have food that's going through our digestive tract, it actually takes about 12 to 24 hours from the time we ate it for it to get passed out as fecal matter. So it stays in that large intestine for a while, fairly slowly moves through it, and this water reabsorption can happen over this longer period of time. The other thing that happens in our large intestine, like we talked about for other animals that are primarily herbivores, they have gut bacteria that help them digest plant matter. We have gut bacteria as well. Now, we don't have an extremely elaborated cecum like herbivores do, but we do have a large population of these gut bacteria at any one time. They do several different things. One is that they do help us digest plant matter, right? Not as much as a dedicated herbivore would. They also do some chemical reactions that our bodies can't do. So they take some of the molecules from our food and they convert them into vitamins that we need and secrete them as waste products, and we absorb those vitamins, right? So some of the work by our friendly bacteria is really important to our overall health. The other thing they do is just by being present in our gut, they outcompete other bacterial species that might be pathogens that might make us sick, right? So they may do a job of actively dissuading those pathogenic bacteria, but simply by being there. So remember, we have competition between different species in an environment. Right, if our friendly gut bacteria can outcompete those pathogenic bacteria for the food that they need, they can exclude them from our gut environment. So we have a community. There's actually many, many, many species of different bacteria that live in our guts. And people have started becoming really interested in this. And so we call it our gut microbiome, all right? The microbes of our gut, like an ecosystem, or a biome, right? Our different plant communities there are around the world. So our gut microbiome is something that's come under a lot of scrutiny lately, right? And so we have discovered, these graphs show, is that where we have patients that have particular diseases or health states, we often find that they have a different community of bacteria in their gut than people who we consider healthy. All right, so this has also been shown to be, we have different gut flora or a different microbiome between people who are mentally well versus depressed, say, or that are obese versus having a healthy weight. Right, so we do not yet understand what the cause and effect is here. All right, do people have these health conditions because they have an unusual gut flora to begin with, or does having this disease cause something to happen in their gut that makes it out of balance compared to what we consider normal? Right, so there are beginning to be treatments for, everything. So when you take an antibiotic medication because you have some kind of bacterial infection, one of the first symptoms you notice is you typically get diarrhea because that antibiotic medication has killed the bacteria that normally make up your friendly gut bacteria. And that causes you to be less able to reabsorb water from your gut because it makes the water just pass through more quickly, right? So if you're taking an antibiotic drug, you can start to help get your gut flora back to normal by taking a probiotic medication, eating yogurt, something that's going to help increase the population of these different bacteria. So a diverse community of gut bacteria in general, just like a diverse ecosystem, tends to be what we see in healthy people compared to unwell people, right? So diversity is good at all different levels. All right, we're going to switch gears now and start talking about the circulatory and respiratory systems. All right, so in vertebrates, we have a respiratory system that's going to consist of gills or lungs, aquatic organisms, gills, terrestrial organisms, lungs. We're going to have a closed circulatory system. So remember that the function of the respiratory system is gas exchange, taking in carbon dioxide, taking in oxygen, getting rid of carbon dioxide. The respiratory system has sort of two component parts that we're going to explore. We have a respiratory surface, right? Respiratory surface is the place where the movement of those gas molecules is between the body and the environment. So we think about absorption happening in the gut, those food molecules or nutrient molecules moving across the cell membrane. Same thing in the lung or in the gill, we have that surface, that place where those gas molecules actually have to go through a cell membrane. This is going to happen by diffusion. We do not have a way to pump carbon dioxide or oxygen across the cell membrane. We're going to have a watery solution on both sides of that cell membrane, and they're going to go down their concentration gradients. The other component to a respiratory system is a method of ventilation. All right, so ventilation is going to be how do we move the air or water across our respiratory surface. And we'll come back to that and how we do it in vertebrates. Our circulatory system is going to be how we move or transport substances around inside the body. It's going to be made of three component parts: a pump, which is going to be muscular and have contractions that force that fluid around. We call it the heart in vertebrates. We're going to have blood vessels. We call them arteries, veins, and capillaries. All right, arteries and veins are large, capillaries are small. And the blood, the fluid that's moving around inside those veins and arteries. We'll come back to those. When we have our discussion of the respiratory system, it's tied to our discussion of the circulatory system, because when we look at how the carbon dioxide and oxygen get taken to the cells, places where they are needed, and how the carbon dioxide gets back to the lung, how do we get rid of it? We have to think about how it's moving through the circulatory system and how it gets delivered to the cells. So even though the little top section of this diagram is really showing the structure of the respiratory system, we've got the respiratory surface here, then we're moving that oxygen into the circulatory system. It's going to go to the heart, it's going to get pumped, it's going to go to the body, right? So these systemic arteries are ones that aren't going to the lungs or gills, right? They're taking that oxygen to places in the body where it's needed. We can also call this the somatic system, right? In contrast, we have the pulmonary system. Pulmonary is going to be associated with the lungs, or we also, in our fishes, we have gills, right? The capillaries that go to the gills. All right, let's look at how do we get air into our lungs, and what's the respiratory surface for humans. All right, so we're going to be able to inhale air through our nasal cavity or our mouth. It's going to go down the pharynx and into the trachea. The trachea is this big tube. It's going to go down and branch to two sides. We have these two big, relatively large tubes that go to our left and our right lungs. And then they branch and branch and branch and branch into tinier and tinier tubules of different kinds. And then the ends of these dead end into these little globular structures called alveoli. A single one is called an alveolus. All right, so these alveoli, instead of being just a blunt end to the tube, they're sort of bubbled into this even more complex three-dimensional structure, which increases the surface area, right? This is the place where the exchange actually happens. All right, so the inside of the alveoli is the actual respiratory surface. The trachea and whatnot are just tubes that are delivering the air to this respiratory surface. We also want to protect these internal structures of our body from pathogens. And so all of our airways are lined with mucus and also with immune system cells that are going to try and grab onto and destroy any kinds of pathogens that are coming in through our airways. All the lining of the surface of these tubings have little cilia on them, and they're constantly going to be beating and moving this layer of mucus in this kind of escalator effect. Any kind of dust or soot or anything that we've breathed in is going to get hopefully trapped in this mucus, moved up and out, and you're either going to cough it up and spit it out, or you're going to swallow it. If you swallow it, it gets digested, right? All right, so these alveoli, there's air inside of them, right? They are connected with the outside world. And as we breathe, we move air into these spaces and then out of this space again. While the air is in there, there's going to be a concentration gradient. It's going to be higher oxygen level than in our capillaries. So oxygen is going to move by diffusion. Now remember, our respiratory surface has a couple of characteristics. One is that it's in contact with the external environment. The other one is that it's moist, right? The oxygen has to dissolve into mucus that's on the surface of the epithelium. If it's a dry cell, it can't get inside of it, right? These are living cells as well. We have to secrete mucus to always keep them coated with a watery layer so that they can stay alive, just like our eyes, right? This epithelium is thin. We want to be easily able to move oxygen from one side of it to the other because what's right behind it, this is another characteristic of our respiratory surface, is that capillaries are right behind it. So we can pass the oxygen straight away into them. All right, so we have these capillaries wrapped around the outside of the alveoli. All right, so this looks like there are all these separate little grape clustery things, but when we actually look at the lung, we slice into it, it really looks more spongy. It's hard to tell all these little tubes apart from one another, and they're really packed together. All right, so that is the basic structure of the vertebrate lung, at least for mammals, sorry, the mammalian lung. Now I want to talk about how do we get air into and out of those spaces. Well, we rely primarily on this diaphragm muscle. So the diaphragm muscle, when it's relaxed, it's dome-shaped. It sits on top of our liver and stomach. And it actually, if we look at it from the top or bottom, it completely seals our chest from our abdomen. All right, so the way our respiratory system works, the way our respiratory system works is that we have a sealed space. It forms what's called our thoracic or chest cavity. The only body parts that are in our thoracic cavity are our heart and our lungs, and they're protected inside of our rib cage. So there is only a certain amount of space around the outside of the lungs, and it doesn't have any air in it. So it's a vacuum. So what happens, right, so remember this is a dome-shaped muscle, is that when we are breathing, we contract that muscle and it flattens. All right, what that does is it makes that space of our thoracic cavity, it increases its volume. All right, physics students, when we increase the volume, what happens to the pressure? The pressure goes down. Now we have a negative pressure in our chest cavity compared to what it was previously, compared to the outside world. And so when we inhale, we're actually sucking air into our lungs based on this negative pressure that we create. Okay, so we consider mammalian breathing negative pressure breathing. All right, so the diaphragm moves down. That makes the thoracic space bigger, makes the pressure in it lower. That causes air to get sucked in, right? It's going down its pressure gradient. When we relax the diaphragm, it moves up, right? It pushes on the thoracic cavity, and also this really neat structure that the ribs have, the shape of them and how the muscles are fastened to them. It means that the rib cage also is getting smaller. So when we exhale, our rib cage is actually squeezing down on that space. All right, so we're going to push the air out by making that chest cavity smaller. And then when we relax those muscles, contract the diaphragm and the rib muscles, it makes the thoracic cavity larger. All right, so in contrast to our system, when we have a diaphragm muscle, right, we look at amphibians. Amphibians, like a frog, when we do our dissection and we can look everywhere, they do not have a diaphragm muscle. They just have one body space, right? How do they get air into their lungs? They do have little stubby lungs. Well, it turns out that the top of those, the tubes that connect those lungs to the outside world is in the back of the mouth. And what they have to do is actually gulp air and swallow it and forcefully push it down into their lungs. So we say that they have positive pressure breathing because they're exerting, by swallowing with their muscles, they're exerting a force that's pushing the air into the lungs. All right, so when we inhale and exhale, we don't actually exchange all of the gas that's inside those spaces. And if you've learned to scuba dive, you would have had lengthy conversations in your class about this air volume in your lungs, because it affects how much air you breathe on your dive, and it also affects your sensitivity towards getting the bends, right? So when we're looking at a patient and we're concerned about their respiratory health, we may do some analyses where we look at how much they're able to breathe on each breath. So there's a series of volumes that we can calculate for a patient, and then the capacity, or these sort of calculated things. So when we're resting and we're breathing our breathing is calm, right? We're not trying to take super deep inhalations or exhalations. That's sort of our normal range of breathing in and out, and we call that the tidal volume, like the tide going up and down. So you can see from this figure that these little small inhalations and exhalations don't represent the entire volume of our lung space, right? In an adult, that amount of air that we could typically inhale and exhale is about 500 milliliters, right? Now, there is some airspace, right, that's dead airspace in our lungs. Even if we exhale as hard as we can't get all the air out, right? That's because we have to inhale it and then exhale it again. And there's always some residual amount that's left sort of in the bottom, right? And that's called the residual volume. So that's space in our lungs. If we filled our lungs full of water and then squeezed it out, which sounds like an awful thing to do, we would find a volume, right? And then we measured the amount of air that we were inhaling and exhaling. We wouldn't get up to that total volume. So there's this sort of leftover, that's the residual. But when we do breathe more deeply in and out, we do get sort of a reserve. We get there's some extra space, right? We can breathe more deeply than we normally do. So we have an ability to take an extra deep breath and inhale, and that's called our inspiratory reserve volume. All right, that's room for extra inhalation if we need it, or our yoga teacher is telling us to. All right, and we also have an expiratory reserve volume. So this is how much we can increase the capacity of our breathing if we want to or need to, right? We can do this consciously. We'll start breathing harder if we have activity that increases our oxygen demand as well, right? So that potential that we aren't always using, but it's there, that's called vital capacity, vital capacity. All right. So mammals have a pretty good ability to extract oxygen from the air, but not as good as birds. All right. So we have basically, if we think about a complete digestive system versus an incomplete digestive system, right? When you take food in, it's an incomplete digestive tract, and then you have to sort of squish the waste products out. You've only got one opening. It serves as both mouth and anus, ick, right? Versus a complete digestive tract. It's much more efficient to have that one-way flow, to have a mouth and an anus. So our respiratory system acts like an incomplete gut. We've got to move the air in, and then we've got to push it out again. Birds have a system that comes very close to having a complete respiratory tract. Now, they don't breathe air in and then breathe it out, some kind of opening that would sort of be like a respiratory anus, which doesn't exist, but they do breathe it back out their nose. But as it is, it goes on a loop so that it's always only going one direction through the lungs. The lungs are static, right? They're not like ours that have to sort of stretch and contract again. And so their structure can be more delicate without being sensitive to damage. That means that they can have an even higher surface area on their respiratory surface that's in their lungs. And that, because their air is flowing one direction through the lung, they can set up their capillaries so that they have counter current flow of the air and of the blood in the capillaries so that they can maximize the transfer of oxygen from the air into the circulatory system. The way they do this is that instead of having the lung be the part that through negative pressure they're sucking the air into, they have several air sacs. They have air sacs that are anterior to the lungs and also posterior. When they first inhale, the air goes to the posterior air sacs. They are posterior to the lungs. Right? So by negative pressure, they're going to inhale, relax the space around that air sac. But then when they squeeze the muscles around that air sac, it isn't going to force it back into the trachea. It's going to force it into the lungs. So there's actually valves that prevent backflow. All right, so they're going to inhale once. That is going to move air into the air sac. Then they're going to inhale again, and that is going to move air into the lung. And the reason it moves into the lung is because the anterior air sacs are also expanding, and that creates negative pressure that sucks the air from the lungs through, and then ultimately it sucks it into those anterior air sacs, and from there it is exhaled. There are also valves that keep it going one way. All right, so these air tubes in the lung have one-way flow so that they can have counter current flow between the air and the blood, which maximizes exchange, like we talked about with heat. So there's a structure that enables them to have associated improvements in function. And that they also have this increased surface area of the air tubes in their lung compared to mammals, because it doesn't have to change it doesn't have to stretch, right? Ornithologists, people who study birds, hypothesize that these mechanisms evolved in birds in their evolutionary history when they were still reptiles. Well, they are reptiles, but before they were the birds we know of today, at a time when oxygen on the planet was about half the concentration that it is today. So that their lungs actually are able to absorb way more oxygen than they actually need. Physiologists who are studying birds can observe their blood chemistry, and basically they never get out of breath. They get overheated, but even at very, very high elevations where the oxygen level is low compared to sea level, the birds are not oxygen stressed, right? It's pretty amazing. People who study birds basically consider them to be, you know, the super sports car version of anatomy and physiology compared to mammals. I've just been learning this stuff recently, and I'm starting to agree. Everything all these mechanisms that I read more and learn more about, I'm like, wow, yeah, that's so more efficient, right, than the mammalian system. All right, mammals are awesome too. Okay, so it turns out that even our poultry system in mammals compared to birds allows us to, for the most part, get as much oxygen as we need. And that's in part because we have a respiratory pigment, all right? Birds do too. It's called hemoglobin. Hemoglobin is stored in our red blood cells. And as our red blood cells are circulated around the body, they're able to pick up oxygen from the air in the lungs because they have a lower oxygen concentration compared to the air spaces in the lungs, and then drop the oxygen off in places in the body that it's needed because the circulating blood has a higher oxygen concentration than the tissues, right? So we've got the lung airspace at the top, we've got the body tissue at the bottom. Obviously, this tissue could be anywhere in the body. This is a very simplified version of the circulatory system compared to simplifying, right? So at sea level, the partial pressure of oxygen, which is measured in millimeters of mercury, is about 100 millimeters of mercury. Deoxygenated blood that's coming from our body tissues is only going to have a partial pressure of about 40. Right, so we have a gradient, we have a difference. So oxygen is going to move by diffusion from the airspace in the lung to the blood. Once it gets in the blood, it gets transported. When it gets to a place in the body that needs it, right, it's not going to deliver oxygen where it isn't needed. It will know that oxygen is needed because the oxygen concentration will be low. Anywhere in the body, the oxygen concentration is lower than the concentration in the bloodstream. Oxygen will be transferred from the blood to the body tissues, to the cells that need it, by the process of diffusion. Now we have deoxygenated blood. It's getting transported, right, back through the circulatory system to the lungs to pick up more oxygen. At the same time, carbon dioxide, which is produced in the body tissues as a byproduct of cellular respiration. We have a high concentration of carbon dioxide in the tissues where it's being made. We have a low concentration of carbon dioxide in the blood compared to in the tissues when it's coming from the lungs. So it's going to move by diffusion from the body tissues into the circulatory system. It will be transported, right, carried to the lung. Higher concentration, 46 millimeters of mercury, partial pressure of carbon dioxide in the blood. It will move down its concentration gradient by diffusion into the airspace of the lung, right? Air from the outside has a lower concentration of carbon dioxide than the carbon dioxide in our bloodstream. So we've unloaded this carbon dioxide, and now the blood that is moving around is going to be low in carbon dioxide. We've offloaded the extra at the lung. We're ready to pick more up. All right, so we're going to be able to literally exchange. When we talk about gas exchange, remember that one thing is coming in and something else is going out. All right, so we're trading gases for one another using the same system. All right, we're able to effectively and efficiently do this exchange in large part because we have hemoglobin molecules that are being carried in the red blood cells. So we have this molecule that's called a respiratory pigment. It's a pigment molecule because it looks a color. It looks red because it has an iron atom in the middle of each of these protein chains. And the iron, if you've ever had anything iron rust, iron oxide, right? So the iron has an affinity or an attraction for oxygen. And when they combine together, they will look red. All right, so our blood is red because of the iron containing respiratory pigment. Other organisms have different respiratory pigment molecules that have different metals in the middle of them. And so their blood looks different colors. Right? There's organisms with blue blood, with green blood. Right, so having this respiratory pigment actually allows us to transport 50 times the amount of oxygen that we could without it. Right? 50 times. All right, why? Why does that happen? Partly because when the oxygen, even if we didn't have it in red blood cells, right, even if the hemoglobins were just floating around, as the oxygen is attracted to and reversibly binds to the iron, it moves out of solution, right? And diffusion is happening from one solution to the other. So when we have oxygen moving into the blood or the plasma from the airspace of the lung, as long as the oxygen is continuing to stay dissolved, all right, we can't move anymore, all right? But the oxygen is binding to the iron. That means it's moving out of solution. That means there's room for more oxygen to move by diffusion into the plasma. All right, now we bundle up a whole bunch of these hemoglobins into red blood cells. We do an even better job of moving that oxygen out of the plasma component of the blood. We have this compartment or partition, right? So many processes in physiology are more efficient when we can partition or separate different components of the body, and we can move molecules from one place to another. This is another example. All right, so each hemoglobin molecule is actually made up of 4 parts. There's 2 kinds of protein chains. So it takes 2 genes to make a hemoglobin molecule. So hemoglobin is a protein with tertiary structure. There are alpha chains and there are beta chains. Each hemoglobin is made of 2 alphas and 2 betas bonded together. And each chain has a place for binding one oxygen or O2 molecule. So each hemoglobin carries 4 oxygens. Each red blood cell has about 1 billion hemoglobins. All right, that's a lot. It's a lot. And then I don't know off the top of my head, but there's probably also 1 billion red blood cells circulating in your circulatory system. So that is part of the reason why we have this really excellent ability to transport oxygen in the circulatory system. When we look at the entire human circulatory system, it actually only takes about a minute for a red blood cell that just got oxygenated in your lung to take a route through the body, drop off its oxygen, and come back again to the lungs and pick more up. All right, some other amazing things about the hemoglobin molecules. Their structure changes slightly depending on conditions. And the changes in their structure determine how strong the binding is between the iron and the oxygen. So affinity is how strong the binding is, right? When oxygen levels are low, the hemoglobin molecule is going to have a higher affinity for oxygen, right? So that's going to cause it to bind better. Now we'll see in a minute that as the body increases in carbon dioxide, that can also slightly decrease the pH. So when carbon dioxide dissolves in water, it releases a hydrogen ion, so we could get slightly more acidic. So it also turns out that a slight acidity is going to increase the affinity of the binding of the oxygen as well. And so that where the body is oxygen stressed, we're going to be able to pick up more and transport more oxygen. All right, so there's also a couple more mechanisms besides just this affinity between the oxygen and the hemoglobin. All right, so the hemoglobin molecule that is contained inside the red blood cell is going to pick up and release the oxygen, and it's going to be able to release it when oxygen is needed and absorb it when oxygen is not, based on those differences in affinity of the hemoglobin molecule. There's a slightly more complicated version of transport of carbon dioxide, right? So we not only want to deliver oxygen, but we want to pick up carbon dioxide and get rid of it as well. So carbon dioxide is going to diffuse from the body tissues that have been busily doing cellular respiration. Right? So we're going to produce it in the body tissues. It's going to move into the plasma. So the plasma is the watery stuff that makes up all the rest of our blood that isn't made of cells. All right, so we've got these red blood cells floating around. The red blood cells have a whole bunch of stuff in them besides the hemoglobin, right, including some enzymes. There's an enzyme that's going to facilitate chemical reaction that is going to combine carbon dioxide and water and form carbonic acid, H2CO3. Right? Carbonic acid is going to spontaneously dissociate. It's an acid because when you mix it with water, it gives off a hydrogen ion. And those bicarbonate ions, HCO3 minus, are then going to diffuse out of the red blood cell. Now, this whole series of chemical reactions is reversible. So if you're learning in your chemistry class that we have certain chemical reactions that are going to get tugged one direction or the other, depending on concentration, right? Solutes become, so salt, substrate is substrate only when it has a higher concentration, and then it's going to get pushed towards reactants. But when the concentration of reactants builds up, it's going to get pushed back towards what we started with originally, right? So these are these reversible reactions that depend on concentration differences. So each one of these arrows here, right, this arrow moving carbon dioxide and water to make carbonic acid, and carbonic acid dissociating into bicarbonate plus hydrogen ion, those are both reversible. And so where carbon dioxide concentration is high, it's going to push this chemical reaction this way. It's going to be helped by enzymes inside the red blood cells. All right. And the hemoglobin helps with that as well. Right. So hemoglobin does not just help carry oxygen, it helps carry carbon dioxide. And the way that it does that is that not the heme group, but the other end of those protein chains that make up the hemoglobin have an affinity for carbon dioxide. So some of the carbon dioxide is actually going to stick to the hemoglobin itself. Another part of the hemoglobin molecule has an affinity for hydrogen ions, right? So those hydrogen ions don't go out into your plasma and make your blood acidic, right? They get stuck onto the hemoglobin molecule. So they stay inside the red blood cell, right? That helps manage homeostasis of pH of the plasma. So this system actually means that most of the carbon dioxide that's carried in your circulatory system is transformed into bicarbonate ion. Right? So the bicarbonate ion is going to get moved throughout your circulatory system So about 70% of all the carbon dioxide that gets transported is in this form. Right? About 23% of all the carbon dioxide that's transported stays inside the red blood cells, and only about 7% stays in the plasma. Right? Again, the same way with oxygen, this means that we can maintain a concentration gradient between the body tissues and the plasma. Right? We're slurping the carbon dioxide up into the red blood cells so that we can absorb more of the carbon dioxide from the tissues, transport it away, because we're accentuating or continuing to allow a gradient to exist in concentration between the tissue and the plasma, right? So these reactions are reversible. So that means that when we get to the lungs, now there's air spaces in the alveoli that have a lower concentration of carbon dioxide than the circulating blood does. And so they're going to start pulling these chemical reactions back in the opposite direction. Right? So carbon dioxide is going to diffuse from the plasma into the airspace. That's going to decrease its concentration. That's going to pull carbon dioxides out of the red blood cells. That's going to cause them to be turned from bicarbonate ion into carbonic acid. The hemoglobin is going to release the carbon dioxide and the hydrogen ion because, again, its affinity changes based on the concentration around it. And that will actually pull bicarbonate ion from the plasma into the red blood cells to get the converted back to carbon dioxide. All right, and that is all caused by this tug, this pull of the decreasing concentration when we start to move carbon dioxide into the air spaces of the lungs by diffusion. All right, I am out of breath, I am exhausted. We're ready to start talking about the circulatory system in vertebrates. How do we get those red blood cells from one place to another? Well, when we look at the fish-like vertebrates, so early in vertebrate evolution, we see we have a closed circulatory system. We have a heart. However, the heart only has 2 chambers. And the way the blood flows around the body is a little different in animals with gills rather than lungs, and also with a 2-chambered heart. So the way the blood is going to flow is in a circuit or a loop. If we look at how it goes, we imagine one red blood cell moving around. In this fish system, there's only one loop it can go, right? It's going to go from the heart to the gills. It'll get oxygenated in the gills, so we're showing oxygenated blood in red, deoxygenated blood in blue. Oxygenated blood is going to get delivered to the system or to the body. It's going to deliver the oxygen. So it's going to go from red to blue. It's going to pick up carbon dioxide. It's going to carry it back to the heart. The heart's going to give it a pump to help keep the whole system flowing. And it's going to go back to the gills. The 2-chamber heart consists of 1 atrium and 1 ventricle. They are separated by a valve that ensures the blood is flowing one direction. As the atrium contracts, it will push the blood through the valve, which opens based on the pressure. As the ventricle contracts and then relaxes again, it's going to cause a low pressure inside the ventricle. It's going to pull the blood from the atrium as well. Right. Then the valve will close when the ventricle contracts. All of our fish-like chordates are going to have this system with 1 circuit and a 2-chamber heart. They do have a lower blood pressure than mammals and birds. They have somewhat less rapid movement of oxygenated blood to the parts of the body that need it. As we look at the evolution of vertebrates, first becoming terrestrial, and then moving into drier habitats, and also becoming Warm-blooded with mammals and birds, we see an increase in efficiency in the way that oxygenated blood is delivered to the body. So we have, starting with amphibians, we have an elaboration of the fish system. We go from having a 2-chambered heart to having a 3-chambered heart. And this happens during development, sort of by a structure folding back on another one. So we see in amphibians and non-bird reptiles that we have a 3-chambered heart. What we mean by a 3-chambered heart is we have 2 atria and 1 ventricle. Now, depending on what reptile group we're looking at, there are some that are fairly separated. The ventricles are fairly separated from one another, to ones that are not very separated. So there's kind of a range across reptiles. Amphibians have some separation of the 2 sides of this one ventricle. And that's important because where we only have 1 ventricle, but we have these 2 circuits, right? So if we look at a red blood cell and the way that it moves through the body of this animal, we've got a deoxygenated red blood cell coming from the body, from its left toe. It's going to go back to the right side of the heart. It's going to be deoxygenated in this atrium. Then it's going to move to this single shared ventricle. But we have blood that is oxygenated coming from, in amphibians, they can get oxygen both through their lungs and through their skin. Right? So we actually have 2 different sets of capillaries that can be delivering oxygenated blood back to the heart. It's going to go from this pulmocutaneous circuit, which is either to the skin or to the lungs, pick up oxygen, come back to the left side of the heart. So we have this mixture of oxygenated and deoxygenated blood that when this ventricle contracts, it gets pumped both to the pulmocutaneous circuit to go to get more oxygen from the lungs and skin, and it gets pumped to the body, right? So this is showing it as being red, this direction, and blue, that direction. But in reality, we have this purple here, right? This purple is this mixing of oxygenated and deoxygenated blood. So there's actually kind of a little ridge in the middle of this ventricle that prevents some of the mixing, right? But we do have not quite 100% oxygenated blood getting pumped to the body and not quite 100% deoxygenated blood going to the lungs and capillaries. All right, so there's 2 loops here. A red blood cell, if it leaves the heart, is going to go, say, to the left lung and going to come back to the heart. Then it's going to get pumped to the body circuit and go around that loop. So a single red blood cell has to go to the heart twice in order to go all the way around the body. So we say it has 2 circuits. All the reptiles, birds, and mammals also have these 2-circuit heart systems. All right, when we go from having a 3-chambered heart to a 4-chambered heart, we have a complete separation between the left and the right ventricles. All right, so we have complete ability to keep that deoxygenated blood going, all of it going straight to the lungs, and the oxygenated blood going, all of it going straight to the body without this mixing. So when we look here at reptiles, there's all kinds of goofy stuff going on. All I want you to know is that they still are considered to have a 3-chambered heart, that they actually are able to do a better job of oxygenating their blood compared to amphibians. So birds and mammals both have a 4-chambered heart, 2 atria, 2 ventricles. They evolved it separately though. So the reptile ancestor that gave rise to mammals had a 3-chambered heart. The reptile ancestor that gave rise to birds had a 3-chambered heart. Mammals and birds each evolved a 4-chambered heart separately. We can see this happening, the way that it's formed during embryonic development happens a little bit differently. All right, so focusing in on the human circulatory system, it's a little more complicated than the last figure. And we're just showing a little more detail in terms of we've got a left lung and we've got a right lung. We have, when blood is moving from the heart, when it's oxygenated, it's going to come from the left ventricle to the aorta. And then the aorta actually splits and we deliver oxygenated blood via these large arteries sort of to the top of the body and also to the lower body. We also have other branches that go directly to major body organs such as the kidneys. Right? So this is still a simplified version of this diagram. A couple things I want to draw your attention to. The systemic part of this circuit, whether it's the lower body or the upper body, we're going to have arteries carrying oxygenated blood and veins carrying deoxygenated blood. However, these terms are defined not by whether they're carrying oxygenated blood or deoxygenated blood, but they're defined by whether they're going towards the heart or away from the heart, right? So arteries are carrying blood away from the heart. They have different structures and functions, right? And they're able to do their job. What does an artery need to do, right, is different than what a vein needs to do. All right, so aorta is the largest artery. It's a blood vessel leaving the heart, so it's an artery. We have blood vessels coming towards the heart, which normally we call veins, right? So this is an inferior vena cava. It's a very large vein, kind of the opposite of the aorta. But notice the lungs, right? So what's happening, we have deoxygenated blood is going to come to the right side of the heart. Now, you may have noticed, Professor, I'm looking at this diagram, that's the left side of the diagram. Okay, but you have to imagine that you are a world famous heart surgeon and you are looking at the body of a patient who is laying on the table in front of you, right? So we define the sides of the heart based on the perspective of the patient. Okay, so person laying on the table, right? My left side of my heart, you're looking at it and it's gonna look as if it's on the right side of the picture, right? Okay, so we're flipped from what you normally would think of. All right, so right side of the heart is going to receive deoxygenated blood from the body. First, it's going to go to the atrium. We always go atrium to ventricle, nicely in alphabetical order. Atrium to ventricle on the right side of the heart. Now, this is deoxygenated blood that's just come from the body, right? The right ventricle is going to pump, and it's going to pump that blood to the lungs, right? Now, these vessels are carrying deoxygenated blood, but where are they coming from? They're coming from the heart, right? So even though they're carrying deoxygenated blood, they are called arteries, right? And very particularly, they are the pulmonary arteries. One's going to the lungs, pulmones in Espanol. And so this is a weird place in the body where we have a thing we call an artery carrying deoxygenated blood, right? So the blood is either going to go, if you're a red blood cell transiting this pathway, you're either going to go to the left lung or the right lung. You're going to get oxygenated, drop off that carbon dioxide, going to go back to the heart. Vessels that are going towards the heart are called veins. Where do we go? We go to the other side of the heart. We go to the left atrium, then we go to the left ventricle. Right, we have to have this two-chambered system. We sort of have this atrium, right? If you go to a big fancy hotel, that big open area in the front with all the windows, it's called the atrium. The atrium is sort of the place where the blood is going to collect, and it allows us to get a forceful one-directional pump from the ventricle, right? Both atria are going to contract at the same time, and then both ventricles contract. There's separation between those two ventricles. So we have all the oxygenated blood coming from the left ventricle to the aorta. When we're looking at somebody's heart health, the most important part of the heart that we're going to look at to see whether they had a heart attack is the left ventricle. All right, so left ventricle is how we're going to get blood to the rest of the body. So its function is very important. All right, so we basically have this atrium so that the ventricle can give a good forceful push. We have this valve here to prevent it from going backwards. We'll look a little more detail at that. in the next slide. All right. So we have this heart cycle. How does the heart beat? What's happening? What are the different things that are going on? How is the pattern maintained? All right, heart muscle, cardiac muscle is actually different in its structure and its function than skeletal muscle. Your heart has to keep beating, contracting 24/7 your entire life. It turns out that it has an internal mechanism that actually can maintain a set pace, has a little sort of a clock mechanism inside of it. You'll learn more about it in your physiology class. It's really cool. So that it's able to keep making these rhythmic contractions. Now, it does take messages from the nervous system as well. So there's a mechanism to set the pace or the beats per minute of the heart. And it needs to be coordinated. We want to have contraction from the atria and then a contraction from the ventricles, with them both going at the same time. Then a contraction for the atria, then a contraction for the ventricles. Ventricles are larger. It takes them a little longer to fill with blood than it takes for the atria. So the timing of these two contractions is unequal. Right? And when you're listening to a heartbeat of a friend, of your dog, of whatever, a recording of it, and you hear that lub-dub, 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 is this asymmetry between the timing of the contractions of the atria and the ventricles. The heart sounds that you hear, right, are actually the sound of the valves closing. When I was a young student, I thought it was the sound of the sides of the heart chambers thumping against one another, but it is not. It is not. It's the sound of the valves closing. So there are valves that occur after the atria. There are valves that occur after the ventricles. The valves that occur after the atria, which are between the atria and ventricles, are called the atrioventricular valves. The valves that occur after the ventricles are called the semilunar valves. They look like they're half-moon shaped. All right. So the closing of the valves occurs because of a pressure difference from one side of the valve to the other. Just after contraction, there's a higher pressure on the downstream side than there is on the upstream side of the valves, so they close. When there's a forceful contraction from the ventricles, it's going to push the atrioventricular valve closed. It's going to prevent backflow. It's going to push against the semilunar valves. They're going to open, right? But then there's going to be a high pressure of this blood that just got pumped on the other side of the semilunar valves, and that's going to close them from the far side. That's going to allow the ventricles to fill. All right, there's another structure that isn't shown on this diagram, and that is what's the part of the nervous system that is going to control what's happening in the heart. And this is managed by structures of the nervous system called nodes. There's a node on the right atrium, and there's also a node in the septum or wall between the ventricles. The node that's on the atrium is called the sinoatrial node or SA node. And the node that is on the septum, separating the two sides of the ventricles, is called the atrioventricular node. So what happens is that a nerve message comes from the brain if we need to increase or decrease the rate of heart contractions. And that first goes to the sinoatrial node, and it triggers a series of atrial contractions that are on this new schedule. We want to have the atria contract and then this little delay before the ventricles contract. That delay is built in to the nerve that takes the message from the sinoatrial node to the atrioventricular node, right? It's a neat thing. They're called Purkinje fibers, and they're a built-in stall technique. All right, some things that can go wrong with the heart: it can stop beating altogether, or it can get on a bad rhythm so that the ventricles and the atria aren't beating in the right pattern. All right, so that shocking of the heart with electricity, or CPR, cardiovascular resuscitation, where you're pressing on the heart, trying to get it to kind of come back onto a muscular rhythm, those can both sort of reset the beating pattern of the heart. So the muscle cells themselves are capable of making contractions. So if you take a heart, if you cut off all the blood vessels and take it out of the body of a vertebrate, it will continue contracting on its own without any messages from the nervous system, as long as it has enough ATP in the cells to keep contracting. Right? Physical manipulation, sort of causing that contraction to occur by pushing on the heart, can actually trigger it to start contracting again on its own in a small proportion of cases. All right, so two other words on this figure: systole and diastole. Systole is contraction, clenching up. Diastole is relaxing. Right? So we have this alternation of atrial systole followed by atrial diastole, and ventricular diastole followed by ventricular systole. Right? If you've heard those terms associated with blood pressure, we're counting the ventricular, right? Because that's going to be what we're going to measure in your arm is going to be based on ventricular, systole, and diastole. All right, like I said, arteries and veins are defined by whether blood is flowing away from the heart or towards the heart, not by whether it's oxygenated or deoxygenated. When we have blood flowing away from the heart, it's going to be at a higher pressure, right? So we've got high pressure blood. We need to have the structure of an artery that is capable of resisting that high pressure. The vein is going to be returning blood to the heart. The blood is not going to be under high pressure. And we also may be needing to help it along, push against the force of gravity in order to get back to the heart. So there's different structural components of arteries and veins that help them do their functions, right? Artery moving blood away from the heart, it needs to be able to resist that high pressure. So we see that it has a very thick layer of smooth muscle and a thick layer of connective tissue to supply structural stability. We see the vein has a thin layer of smooth muscle and a thin layer of connective tissue. They're kind of big and floppy. If we look at a cross section of a vein compared to an artery going to the same body part, say the vein and artery that are feeding your arm next to one another, we see that the vein is larger in diameter, and we see that the artery is smaller in diameter. And you can see the wall is thicker as well in that picture. All right, we have another structure in the veins that doesn't exist in the arteries. So veins have valves. So valves are going to work just like they do in the heart. They're going to prevent backflow. So if you imagine a large blood vessel of the blood returning towards your heart in your lower legs, say, it doesn't have as much pressure. It's not flowing as fast, and it's moving against the force of gravity. And so where we have, we take a step, and that muscular contraction in our leg is actually going to help push the blood up in our leg. And then it's going to get above one of these valves, and then it's going to sort of close that valve behind it. And that means at the next contraction of a muscle in our leg, it's going to get pushed further up, and it can't flow backwards down and pool in the feet. Now, the aorta, the vena cava, the large veins, large arteries, even small venules and arterioles, is not a place where oxygen nutrients are getting delivered to cells of your body. They're like highways or major roads, right? So the place where the actual exchange is going to happen between cells is in the capillaries. Capillaries are just little tiny blood vessels. They're so small that they're made of a ring or a tube of single cells, right? So they're a thin, the cells are actually curved in order to form into this tube shape. So they're only an endothelium or an epithelium on the inside, right? And a layer of connective tissue on the outside. So when we're imagining oxygen getting delivered or oxygen getting picked up in the lungs, that's happening at the capillaries, right? It's not happening where look at how thick the artery wall is. We're not doing any exchange here. Capillaries also have a variety of mechanisms that help them make sure that oxygen and nutrients get delivered. They may have little gaps in between the cells that actually help fluid get forced out and perfuse into the tissues that need them. And capillaries are grouped together into little bundles. Right? So we'll have this set of capillaries, we'll call it a capillary bed or network, and it's got blood flowing into it, then it disperses into all these little capillaries, and then they fuse together and join together into a vein on the other side. So the way the system is built, we think about as blood is flowing into from an artery into this capillary network, those capillaries fuse back together again to form a vein on the other side. The actual exchange is happening in this purple zone, right, where we're transitioning from being oxygenated to being deoxygenated, transition from being low in carbon dioxide to high in carbon dioxide, to drop off nutrients, pick up waste products, all the things that need to happen. Although we are going from bigger blood vessels to smaller blood vessels, what happens is we add up the cross-sectional area of all the capillaries. It ends up being larger than the blood vessel that it came into that network with, right? So we want to look at the effect of moving through a capillary bed has on 3 variables. And these variables are the area, the cross-sectional area of the circulatory system at that point. And then what the effect of that is on the velocity or the speed the blood is flowing and the pressure. Velocity and pressure can be separate from one another. Pressure is the force that the fluid is exerting outward onto the wall of the blood vessel, and it's measured in millimeters of mercury or pounds per square inch, something like that. All right. So if you imagine that you're standing in the backyard on a nice hot summer day and you've got the hose and it's water's running out of it at a certain rate, and you're like, I want to water that plant that's way over there. You put your thumb across that, partially covering the edge of the hose opening. And what you'll do is increase the velocity of the flow of the water because it's contracted the area that the water is going through. And so it'll squirt faster, which means it'll go farther. Right. So what's happening when we go through a capillary bed isn't that, it's the opposite. Right. So we're going from having a small cross-sectional area to having a large cross-sectional area. What's going to happen? The rate of flow is going to slow down. Right. The rate of flow of blood is going to slow down in the capillaries. So we see that on the second panel. We start with blood flowing rapidly. When it leaves the heart, it's through the aorta, through the arteries, gets down to the capillaries, and it's not going fast at all. It's going really slow. That also helps exchange happen. Because if we have blood zooming through, there isn't much time for that exchange to happen. It's kind of slowing down, it's pooling in the capillaries, there's exchange happening. Now, as those capillaries join up with one another again, the cross-sectional area decreases. So this is where we're putting our finger over the hose, and we're actually getting it to sort of start flowing at a faster rate again, which is going to allow it to get back to the heart. All right, the pressure that the blood is exerting is going to just systematically decrease from the time that it's going to be the highest when it's under the force of that pump from the left ventricle. When it's close to the heart, we in fact will actually see a difference in the blood pressure between ventricular systole and diastole. So that's the difference you see when you get your blood pressure reading. That's why there's two readings, right? There's friction of the flow of the fluid against the inside of the blood vessel. Remember that the arteries have a very thick muscular wall, and they're going to be able to keep the pressure high and basically squirt the fluid through instead of letting it expand in volume and reduce its pressure. But as we keep going through the circulatory system, it's just going to continue losing that pressure, ultimately having lower and lower as it goes through the and coming back to the heart. It's still flowing, it still has velocity, but the pressure is very low, right? You don't want to try taking somebody's blood pressure in their vein. That's not very helpful. All right. So let's look at what happens in the capillaries. All right, one thing about the capillaries is that they have these little upstream sphincters. So remember, a sphincter is like a little drawstring, a muscle that goes around the opening, and when it constricts, it stops the flow, just like in our digestive tract. So we can divert blood flow into or out of capillaries. So when we talk about doing thermoregulation and say diverting blood flow away from your body surface if it's cold outside so the blood doesn't get cooled, this is how we do it. We simply redirect the blood flow to other places. Eventually, those tissues need to have blood flowing through them in order to get oxygen delivered. If we're in a very cold environment and we're in it long enough, there will be a sort of a systematic and regular return of blood flow to those structures. People I know that who've done science in Antarctica says you're just cold, you're out there on the ice, you're wearing this warm clothing, but you're just cold. And every once in a while, you'll get this sensation of warmth of the blood flowing through those extremities, delivering them oxygen so the tissues don't die. Must be weird, never experienced it myself. All right, so what is happening actually in the spaces next to the capillaries? How are we going to get nutrients and oxygen delivered? How are we going to get stuff into the capillary? So a couple of physical mechanisms that happen. All right, so we have pressure coming from the arterial end. So we've got this pathway of blood flowing through this capillary. We have this interstitial fluid. Right, that's the stuff that's filling up the spaces around the body tissues. We have cells that need stuff here, or we need to get rid of stuff. As we go along the length of the capillary, what's going to happen is we have the highest blood pressure at this arterial end. So the graph below is showing what's happening with blood pressure. Blood pressure is going to be going down, right? If we're sort of pushing fluid out at this left-hand end, then we're going to be accumulating fluid in this interstitial space. That means it's going to flow back in, right? So we're having a lower blood pressure at the venous end of the capillary. That's going to suck fluid in, right? And that is some diffusion is going to happen as well. Diffusion is going to get how we have substances actually absorbed by the body tissues, but there's actually this bulk flow moving into and out of the capillaries as well. We also are going to have at the downstream end any materials that have a higher concentration in the interstitial fluid are going to move into the capillary and vice versa. All right, we've talked a bit about what's in blood, All right, we're going to go into a little bit more detail here. So blood is considered a tissue. It's made up of cells or cell-like components and watery component. The watery component is mostly water, but it has many, many, many dissolved substances and solutes. So the watery component is called plasma. If by volume, it's about 55% of a blood sample. The cellular elements, including white blood cells, red blood cells, and these other little structures called platelets, which aren't cells, but are cell membrane based, make up about 45%, 46% total of the volume of your blood. Right? So there's this liquidy stuff called plasma, and then cellular components. Something unique about our circulating blood cells is that we continue to generate them throughout our lives. They come from stem cells in the bone marrow. Stem cells in the bone marrow are going to be able to create two different kinds of cells. The lymphocytes are part of our immune system, or being T cells. They're a special kind of white blood cell. The myeloid stem cells are going to generate all of the other kinds. So they're going to differentiate into all of these other kinds of white blood cells, also red blood cells, and again, platelets. Red blood cells' main job is transporting oxygen and carbon dioxide. White blood cells are part of the immune system, and platelets are important in clotting. We'll talk about this clotting mechanism, right? Blood clots in your brain are bad. A blood clot, fixing a hole in your circulatory system is good, okay? So other stuff in your plasma, so it's water, and it's everything you can think of that you can get a blood test for and more, right? We're gonna have glucose and other sugars. We're gonna have oxygen, carbon dioxide. We're going to have hormone molecules. We're going to have all kinds of proteins. We're going to have waste products like small amount of ammonia and urea. We're going to have chemicals that have been introduced into your body, such as drugs or medicines. And we're going to have a whole bunch of dissolved proteins that serve different functions. Besides platelets, there's a bunch of other proteins in the plasma that participate in forming clots. When a blood sample is taken, it can be analyzed either by letting it naturally clot, which it's going to do when it comes outside your body, and then removing the clotted stuff, in which case you're left with a liquidy substance called serum. All right. So the difference between plasma and serum is that plasma has these proteins in it that participate in making the clot. And in order to keep it like that, an anticoagulant molecule has to be added to that sample. Right? So for different reasons, we would analyze blood samples in these different ways. All right, so we want to have the ability, when we have damage to a blood vessel, circulatory system structure, to be able to repair it. Okay, we live by having an intact circulatory system. Remember, it's a closed circulatory system. We especially don't want it to be open to the outside world and be squirting blood all over the place. All right, so we have a mechanism that is going to be able to form a structure called a clot, right? A clot is going to be a mixture of platelets. So platelets are cell membrane. It's in kind of these little cell fragments, and they've got little kind of pointy edges. Then there's other components of the clot, and one of them is a molecule called fibrin. So fibrin is going to form these sticky threads. The sticky threads are going to capture red blood cells. They're going to stick, right? It sort of makes like a spider web. And the platelets are attracted to places where there's damage to the internal structure of a blood vessel. How does it know there's damage to the internal structure of the blood vessel? Well, normally epithelial cells lining the inside of that blood vessel are not going to have any fibers of the molecule called collagen exposed at the surface. If there's damage to the blood vessel, these collagen fibers will get exposed. The platelets can detect and bind to the collagen fibers, and they get triggered to release chemicals, some kinds of chemical messengers, sort of like hormones. They're going to do a couple things. They're going to make the platelets sticky. They're going to attract other platelets, and they're going to trigger a set of chemical reactions that happen in the circulating bloodstream. So remember, we talked about precursor molecules. You don't want to have clots all over the place all the time, because we know that when we get a clot in the wrong place at the wrong time, it can clog an important capillary, like one going to your brain, and you have a stroke and it could be fatal. We want to have a clot form only in the places where there's damage that it needs to form. But we want to be able to form it rapidly. So we have these precursor proteins circulating in the bloodstream all the time. They're not sticky, they're not going to form a clot, but when they're triggered to, they will, okay? There's actually a series of about 10 molecules. This one is only showing prothrombin getting turned into thrombin. Thrombin then catalyzes the conversion of fibrinogen into fibrin. Fibrin is these sticky threads. Right? So we have these damaged cells secreting molecules that are going to tell nearby platelets to come close by. This cascade of conversion of clotting factors is going to occur. The disorder, the blood disorder hemophilia, where clotting doesn't happen, can actually be caused by a genetic mutation in any one or more of any of these clotting factors. So there's a variety of different forms of it. All right. So we get the sticky threads, we get the platelets, we get some blood cells stuck in there, and you may think, doesn't sound like it's really going to do a good job of plugging up this hole. Well, certainly if the hole is small, yes. If it's a very large hole, no, this isn't going to work, right? So that's why you have to go to the doctor and get those stitched up, right? If you get it sewn together tightly, then the clot will work on that small location. But if you imagine how, if somebody in your family has long hair and they go into the shower and some of their hair comes out when they're washing their hair, and that hair gets into the drain, it doesn't actually take that much hair to clog the drain completely, right? So this is not only hair, this is like grabbing other stuff, and it's sticky. So it's really, it does the trick, right? All right, shifting gears, we are going to wrap up our survey of vertebrate organ systems with the kidney. Two main functions of the kidney. It's part of the osmoregulatory and excretory system. So it's going to produce urine. That's its job. What is urine? We've filtered the blood. So we've taken some of the plasma from the blood, moved it into this little tubule, this excretory tubule. In the vertebrate body, these tubules are inside the kidney. Right? So imagine we're inside the kidney. We have these little tubules. They're going to be called nephrons. The nephron is going to have some of this plasma moved into it. The first step in moving the plasma from the capillary into the nephron tubule is called filtration. Why is it filtration? Because we're leaving the cells behind. We're leaving the red and white blood cells behind. We're only moving the plasma. Imagine you're filtering your coffee, or you're filtering some precipitate in chemistry. The bigger chunks stay on the filter. The liquid or the supernatant moves through. Same thing in coffee. The grounds, the chunky bits stay on the filter. The liquid only, no crunchy bits, go into the coffee and all the dissolved substances. This delicious flavor molecules, caffeine. All right, so the concentration of stuff, all the stuff that's not cells, is the same in plasma as it is in filtrate. There's stuff in there we want to get rid of. There's stuff in there we want to keep. So we have these two processes that are going to go on more or less simultaneously as this material, the filtrate, moves through the nephron tubule to the point where it's finished, right? It's restructured to its final product. We'll call it urine then, right? We want to take some materials back from the filtrate, right? We're going to say we're reabsorbing them because they're going to go back into the capillary. Now there's other molecules we might want to actively secrete, right? Some of them is going to go into the filtrate, but we want to get rid of more of them, right? In that case, we can do secretion. So we will use ATPs to do active transport and move those additional amounts of them into the nephron tubule. Right, so this is the basic idea of what's going to happen in the nephron tubule. The first step is filtration, right? Then we're going to balance out the stuff we want to keep and get rid of. And then the final product, the urine, is going to go out of the body. All right, so humans have a pair of kidneys. We're bilaterally symmetrical. We've got one on each side. Occasionally, people are born with only one. You can live with only one if you donate your kidney to a relative. And the whole excretory system is pretty simple. It only has 4 parts. It has the kidneys. It has these tubes called ureters that drain the urine from the kidney into the urinary bladder. Urinary bladder is a place where it's temporarily stored. We have control over when we release it and when we don't release it to the outside environment from a sphincter, just like we do for fecal matter. And then there's this tube that connects the urinary bladder to the outside world called the urethra. In female mammals, the urethra has its own separate opening. It's separate from the opening for the digestive tract, the anus. It is separate from the opening for sexual reproduction, the vagina. In male mammals, the urethra is a shared reproductive function. So it is the tube that goes out through the penis. It is used for urinating. It is also used for passing the sperm and the mixture of other components that leave the body during ejaculation and allow our species to have internal fertilization. All right, don't get the ureter and the urethra confused. Figure out a way to remember it. All right, so when we look at the kidney, if we take a slice down through it, the kidney bean got its name because it's the same color and shape as a kidney, not vice versa. We're going to see that there's actually a different color of the tissue that we can see when we slice through it, and that the different regions of the kidney are different colors because they have different structures and different functions. The outer region is lighter in color. It's called the cortex. The inner region, or the middle, is called the medulla. We have different things that are happening in those places. So this whole kidney has, let me look at my notes, how many? It has about 1 million nephrons in each kidney. Okay, more kidney fun facts. About 25% of the blood leaving the heart goes to the renal artery. All right, so it gets processed in the kidney. So often diseases or things that relate to the kidney are called renal, R-E-N-A-L. That it's a relatively small organ located kind of in the small of our back. It's about the size of a fist. It does a lot of active transport. So it's a highly energy using body organ. Our brain is the highest energy using organ in the body. The kidney's up there. It filters our total blood volume in only about 40 minutes. So if you think about that cup of coffee that you drank and then how soon it is when you go and pee that you can smell the coffee or whatever other strong smelling foods that you might have consumed, that is the time it has taken for some of those molecules to get absorbed from your stomach and your intestine, get transported through your circulatory system, and enough of it to get filtered out of your blood and passed in your urine. It's pretty amazing. And we require a total of about 500 milliliters of water a day minimum to make sure that toxic molecules that are in our circulating bloodstream are getting sort of rinsed or flushed out of our bloodstream via our kidney. So one way that mammals have adapted to different environments is the proportion of their kidneys that is devoted to different aspects of the work. So where we have this renal medulla, we'll see that some of the nephrons have this long loop that passes down deep into this region. And those particular nephrons are how we can concentrate solutes into the urine. So if we look at humans, there's only about 15% of those nephrons. But if we look at a desert animal that requires to be much more stingy with its water loss, we'll see that a much higher percentage of its nephrons are going to be that long kind that really allow it to magnify the ability to reabsorb water. Mammals that have to drink salt water, like sea otters, have a different structure of the kidney altogether. They don't just have a pair of little like bean-shaped kidneys. They have much larger ones that have multi-lobes and actually increase the total number of nephrons that they have in their body. They can drink salt water, and they have the ability to filter out enough salt that they are able to maintain homeostasis. All right. So we're going to look at how the kidney does its job, and it's pretty complicated. You will likely need to go through this a few times before you understand it. When I was learning it, I was so confused because there's a couple different things that are really happening all at the same time, and you have to know this one thing that's happening at the end of the process in order to make the thing that happens at the beginning of the process make sense. So there's really no way to go through it the first time and have it make sense, but I encourage you to think it through, read it over in the textbook a few times, listen to the video a few times, ask me a gazillion more questions in lecture, because the whole thing works. This is the one sentence summary. That salts, principally sodium ions, are pumped in certain areas to establish high concentration gradients that then pull water either into the tissues or into the tubule itself, right? And we have to do a whole bunch of work to establish a high concentration of salts in these tissues that are very deep in the medulla to allow the reabsorption of water at the end of this process in order to make the urine more concentrated, right? The other thing that's going on is that all these processes are regulated, so that although we do a certain sort of part of these steps kind of the same all the time, there's a whole other section of the nephron tubule that then is able to, based on hormonal instructions, adjust so that homeostasis is maintained. Does the body need a little more salt? It can pull it back there. Does it need a little more water? It can pull it back there. Okay. All right. So we've got all kinds of stuff going on. Give yourself some time and some space to wrap your head around nephron function. This is our last topic in this incredibly long lecture. All right, so let's name the structures first. What are the parts of the nephron? It has a part where filtration is going to occur. And it's hard to see on this diagram, but the Bowman's capsule is part of the nephron. We have some capillaries forming a little network, right, that's coming from a little artery. And the Bowman's capsule is the dead end of this tube, but it fits around like a ball around those capillaries. Why? In order to increase the surface area of that connection. What's happening at the Bowman's capsule is filtration, right? So we're moving plasma from the capillary into the nephron tubule. Then there's a section of the nephron tubule called the proximal tubule. What's it proximal to or close to? It's close to the Bowman's capsule, right? It's wiggly. Sometimes it's called the proximal convoluted tubule, right? Then we have this structure that looks like a hairpin or a road with a sharp turn and then going back almost in the other direction. And it's called the loop of Henle, right? Henle spelled Henle. And it's got a part that goes down. It's called the descending limb. All right, remember a limb is like an arm or a leg. So this is the part of it, the arm that goes down. The ascending limb is the part of it, the arm, the side. It's going up relative to the medulla versus cortex. Then we have another squiggly part, right? This is the opposite of the proximal tubule. The opposite of proximal is distal, right? So it is the distal or far. What is it far from? It's far from the Bowman's capsule. The distal convoluted tubule, it just means it's wiggly. And it's going to drain into a structure that's called the collecting duct. When I was first learning this, I was outraged that the name of this is the collecting duct because it makes it sound like it's not doing anything. It's just collecting, right? That the urine is draining into it. All right, it turns out it's not urine yet. This collecting duct is doing all kinds of stuff. And it isn't until it gets to the ureter that it's urine, right? So it's still filtrate here. It's being perfected. It isn't urine yet. The collecting duct is doing much more than just collecting. All right, we have associations with capillaries throughout the length of this tubule, right? So important to remember as we are looking at the next few slides that as we have blood flowing down this loop of Henle, or sort of down this capillary bed, which is called the vasa recta, and then to this, the other side, it's flowing in the opposite, it's making blood flow in the opposite direction to the filtrate that's flowing through the loop of Henle. So we have countercurrent flow here, right? So between the capillaries and the filtrate in the nephron, we have the ability to maximize diffusion and movement from one of these tubules to the other, right? We also have capillaries closely associated with both the proximal tubule and the distal tubule. That means when we have stuff going into and out of the tubule, for the most part, you need to remember that it's going to be going into or coming out of a capillary. It's going to get whisked away, right? Some of the movement that's going to happen is actually going to move solutes into the tissues, and they're going to stay in the tissues, right? But water that gets moved out of the tubule is going to go into the capillaries and get removed from the area. So that's important to know. All right, so we've got the names of the structures. Now we're going to see that they have different jobs that they're going to do, right? Different names because different things are happening in these different parts. All right, so there's a series of slides here that are kind of showing stepwise how the filtrate is being modified in these different regions. So basically, to summarize again, as quickly as possible, that we're going to have, for the most part, regions where sodium ion and also chloride ion, we care more about the sodium ion. Sodium ion is going to be pumped out, active transport, and the water is going to follow it, right, by passive transport. So notice the difference in the color of the arrows active transport is going to be these orange or red arrows. So that means energy is being used. We have transport proteins that are using ATPs in order to force this solute to get moved across the cell membrane. And then once we've established a concentration gradient, or we're moving those solutes, the water is going to follow. All right, we can't pump water. We have to pump solutes and let the water follow them by osmosis. Right? Okay. Active secretion is going to happen, right? Especially in this figure, we're highlighting secretion of hydrogen ion. That means the cells that are surrounding the capillaries are going to be pumping hydrogen ions. They're using energy to put them into the tubule. That is going to be to try and get rid of those hydrogen ions because they make the circulating blood acidic, and we want to maintain homeostasis of pH. All right. So we're going to have in the proximal tubule a bunch of stuff happening, right? And we're going to have in the distal tubule what looks like almost the same stuff happening, right? It turns out the stuff in the proximal tubule happens pretty much the same time, the same exact way all the time. The stuff that's happening in the distal tubule is under the control of hormones, and it is doing in order to adjust the concentration of solutes and water in the urine based on the body's needs to maintain homeostasis, right? So stuff that needs to happen all the time, proximal tubule, stuff that needs to happen based on homeostasis, needs right now, distal tubule, all right? We're also going to do a whole bunch of work to establish a high concentration of solutes in the inner medulla. Outer medulla is going to have an intermediate concentration of solutes in the tissue. And the concentration of solutes in the tissue of the cortex is going to be about the same as the blood. Why do we need to do a bunch of work in order to do that? Well, because we want to use it to do some other work. All right, so notice down here in the collecting duct that is not sitting there doing nothing, it is actively doing something. Right, that we are going to be able to reabsorb water from the urine into this inner medulla, right? And the reason we can do that is because we have done a bunch of work, which isn't shown on this slide, to get concentration of ions into this tissue. And so if we were going to take this live, fresh kidney and we would eat it, would take a bite of it, right? It would be really salty on this internal part, right? So we're doing active work to put salts into this tissue here, and that is going to allow us to suck the water out of the collecting duct if we need to, right? If we are dehydrated. It's not going to go into this tissue, it's going to go into a capillary so that it gets reabsorbed and back into the bloodstream. Right? Because if we just let it go into the tissues, then it would dilute that concentration of solutes, plus we'd swell up and it wouldn't be good. All right. How do we manage to do this? Okay, so there's a couple mechanisms. Different parts of the tubule are going to allow different things to pass through the wall of the tubule. So they're going to be permeable or not permeable. Different places are going to be allowing water to go through them or not allowing water to go through them, right? So we'll look at different parts of the tubule and look at what's different about the parts of the tubule. All right, so there's going to be things that we want to do all the time. We want to pull back much of the water from the filtrate most of the time. So we're going to actively be pumping sodium and letting the water follow. We don't want to pee out nutrients. We don't want to pee out excess, we don't want to pee out glucose. Right? So one of the things we're doing is we're actively reuptaking nutrients like glucose. So what we're going to do right away is cause some of this water to be withdrawn from the filtrate. That's going to cause the volume of the filtrate to decrease a little bit, right? So as it's going down into the tubule, the volume of the filtrate is decreasing. There's also another mechanism that the structure of the tubule can use as part of its bag of tricks. There's a protein, a transport protein called an aquaporin, right? An aquaporin, we still can't pump water from one side of a cell membrane to the other, but we can open little tubes in the cell membrane that cause water to be able to flow through it much more rapidly. All right, so we have lots of these aquaporins in the cell membranes where we want to move water across the cell membranes easily. So for example, we have a lot of aquaporins in this region of the tubule where this water is moving out. Now this sodium is getting pumped out actively on this other side over here. Why isn't water following it? Because we have no aquaporins. In fact, this tubule is sort of reinforced to make it extra impermeable to water, right? So we don't want water to follow the solutes in this portion, right? So on the ascending limb of the loop of Henle, it's not permeable to water, whereas on the descending limb, it's very permeable to water. All right, and we'll see another neat mechanism for how we recapture a lot of water rapidly, but only as needed in the collecting duct in a couple of minutes. All right, so we're gonna look through a couple more slides that basically show the same thing, but show how concentrated solutes are in these different tissues and how concentrated solutes are in the filtrate. So we start with a concentration of solutes of about 300 milliosmoles per liter in the plasma and in the filtrate. All right, that concentration doesn't change too much as we go through the proximal tubule. But when we start getting into the descending limb of the loop of Henle, notice that water's going out, right? Why is that happening? Because we have done a bunch of work somewhere else to have a higher concentration of solutes in the tissue. Right? So we have from 400 to 600. It's increasing, the concentration of salts it's increasing as we go deeper into the inner medulla. So as our filtrate is flowing down the descending limb of the loop of Henle, water is getting pulled out of it by the concentration of solutes that is in the tissue. Where is it going, right? It's going into the capillary that is wrapped around the loop of Henle, right, called the vasa recta. And it can do that because that capillary has a higher concentration of solutes than the other end. It's pulling the water in as well. All right, so what happens is we're decreasing the volume of our filtrate and increasing the concentration of solutes. We can't increase the concentration any higher than the concentration of the solutes in the tissue. Right? So we get to about 1200 milliosmoles per liter. As it turns around and starts going back up again, right, now we have this very concentrated filtrate. It's going to be easier for us to move some of the solutes out of the tubule now that it's very concentrated. All right, so we'll see that on the next slide. All right, so because of the structure of the vasa recta and how it's working, we're actually going to be starting to move sodium out of the ascending limb of the loop of Henle. But when we get into this upper part, this part of the outer medulla, this part of the tubule has many, many transport proteins, and it's actively pumping sodium chloride out of the tubule, right? So the volume, but it's not letting water flow after it. So we're actually putting salts into the tissue here. Right, so that explains how we get this higher concentration of solutes in the outer medulla. We haven't explained how we get it into the inner medulla yet, so stay tuned. All right, so what have we done? We have now this smaller volume of filtrate, but we have re-diluted it, not by adding water, but by pumping salts out. All right, so now we have a small amount of dilute filtrate. It's going to pass through the distal convoluted tubule. As it starts moving down the collecting duct, all right, we're going to see how we reconcentrate it. All right, so this work that we've done to pump the sodium out into the tissue, we're going to use it. We're actually using that over here in the descending limb, right? So why was this water getting sucked out of here in the first place? Because the sodium was getting pumped out of the tubules over here. Now you have to keep in mind that there's all these tubules and they're all next to one another, right? So they're all doing the same thing at the same time and creating the overall environment of the tissues of the kidney throughout. All right. So we have this movement of sodium out into the tissues from this very highly concentrated filtrate in the loop of Henle. So that helps to establish the concentration of solutes. The concentration of solutes is also maintained not only by salt, but by urea. You're thinking, wait a sec, we're trying to get rid of the urea. Well, it's not very toxic, right? So we actually have some of this urea is going to diffuse out of the collecting duct. It's going to help keep the concentration of solutes in the tissue high, and it's sort of going to keep cycling around in here. All right, now notice what we're also doing in the collecting duct. We're continuing to actively pump sodium out, and the water's following. The water's going to those capillaries, and so it's getting reabsorbed rather than getting diluting the concentration of solutes in the tissue. All right, so the function of the kidney is really pretty amazing. The ability that it has, the mammalian kidney, to reabsorb almost all the water, about 99%. We can reabsorb 99.5% of sodium ions if we need them, 100% of glucose molecules in a normal healthy metabolism. Urea, we are gonna end up with about 50% of it secreted from the circulatory system. So like I said, we don't actually get rid of all of it. And we can reabsorb for things that we need for homeostasis, for example, phosphate, we can reabsorb 100% of it from the filtrate if we need it. All right. So, for example, growing children or mineralizing the skeleton, repairing a bone after a breakage, something like that. We're able to secrete or get rid of a tremendous amount of hydrogen ion. So actually the pH of urine is a little acidic. It's about 6 rather than being neutral. We're able to secrete almost 100% of many different kinds of organic toxins and molecules, for example, phenol, to get out of the body. Some of that's done by the work of the liver. Other things that are going to end up in our urine are hormone molecules. They're filtered out. They're especially small water-soluble ones. So things where we have a hormone response, things get moved out of the body in the urine if we need more of that, we have to keep secreting more of that hormone if we keep needing it. Other things that we take as medicines or as recreational drugs, they're going to end up getting detoxified, they need to be, or secreted in the urine. So why do we have to keep taking a medicine over and over again every day? It's because the body is actively breaking it down and getting rid of it, right? So we need to keep taking more and more. So our kidneys actually do an amazing job of keeping our bodies safe and free from harm and regulating amount of work that they do in terms of homeostasis of our circulating bloodstream. It's just astronomical. All right, thank you, kidneys. So when we look at what's happening across the pathway that the urine takes as it goes through, we have some stuff happening kind of over here and in the middle that's making the work done possible that's over on that left-hand side. All right, so we have to understand all these different parts in order to see how all the parts work. Right, so hopefully you go back again through it, it'll make a lot more sense the next time. All right, our last topic is, okay, so we're doing homeostasis of water balance. How do we do that? So a couple neat things about regulation of water. It turns out there's two different problems we can have physiologically. One is that we can just be dehydrated. We don't have enough water. The second one is we could be not just missing water, but also missing solutes. All right, so we could be dehydrated just if we're sweating a lot. It doesn't really mess up our entire our concentration of solutes, or we haven't drank very much water. If we have severe diarrhea, if we have lost a lot of blood, we've not only lost the water, but we've lost the solutes. All right, so two different problems. We have two different hormone systems to fix them. All right, so we have osmoreceptors in the hypothalamus. They're going to trigger the release of antidiuretic hormone. Also going to make us feel thirsty. If we're thirsty and we drink water, then that will restore our blood osmolarity to the correct amount. But in case we don't have that option, or we don't listen to it, our pituitary gland is going to secrete antidiuretic hormone. A diuretic makes us pee. Antidiuretic hormone makes us pee less, right? Or resorb more water. What it's going to do is trigger the reabsorption of a lot more water in the collecting duct. So what we see here on this panel to the right is our ADH receptor. Now, remember, our hormones have to have a receptor that's at the surface. It's going to trigger something. In this case, it's CAMP, cyclic AMP molecule. It's going to send a message, couple steps. And what that's going to trigger inside the cell is that these vesicles are going to fuse with the cell membrane, right? They're going to do exocytosis, right? So they're storage vesicles, but this is a sneaky thing. We don't really want the stuff inside of them to get moved to the outside of the cell. We want the vesicle itself to get fused with the cell membrane of the outside cell, because what is in the cell membrane of all of these vesicles? Aquaporin water channels. So we are going to make the tube, the collecting duct tubule, we are going to make it more permeable to water by suddenly and rapidly shoving a bunch more aquaporins into the cell membrane. When we've got enough water, we're going to do basically endocytosis. We're going to take those little bits of membrane with those aquaporins, and we're going to move them out of the cell membrane. That's going to reduce the permeability. All right, so the collecting duct has the ability to increase and decrease its permeability to water depending on our body water condition. And it has this neato mechanism for doing it. All right, the other thing that can happen, we have a loss of blood volume, right? And body water and solutes as well. We're going to trigger this other mechanism. All right, so we have low blood volume or low blood pressure. That's how our body's going to detect it. It's going to cause release of a hormone called renin. Right, renin gets secreted by a structure in the kidney, and it is going to cause a transformation of a precursor hormone, angiotensinogen, and it's got that gen suffix. So we have a cascade, a series of hormones that are going to cause the release of other hormones. So renin is going to trigger the transformation of angiotensinogen into angiotensin 1. Another molecule is going to trigger the transformation of angiotensin into angiotensin 2. Angiotensin, all right, is going to cause our arterioles to constrict. It's going to make them tense. What does that do? In the short term, it increases our blood pressure, right? It makes the arteries skinnier, decreases the volume, increases the pressure. Doesn't help us if our blood is squirting out all over the place, but in the short term, it increases our blood pressure. Angiotensin is not only going to cause arterial constriction, but it's going to go to the adrenal gland and trigger the adrenal gland to release aldosterone. Aldosterone is going to act on the distal tubule in the nephron to increase both water reabsorption and salt reabsorption. All right, so that's the response when we need to increase not just the water amount, but water and solutes. All right, so it's going to cause sort of this physical reaction, the change in diameter of the arteries to help increase blood pressure. That doesn't solve the total body water and solutes problem, but it's also going to trigger increased reabsorption by active transport. All right, so we're going to upregulate, or we're going to turn on the active transport that's happening in the cells of the distal tubule. Over a longer time frame, that is eventually going to, hopefully, if we have it available to us, increase the blood volume, the blood pressure, and the solutes. All right, and we'll eventually relax this constriction of the blood vessels. All right, so we sort of have a short-term fix and a longer-term solution. Many of our physiological mechanisms have both of those processes. All right, so some of the examples we saw today are of things that mammals and in general have as advantages over amphibians and reptiles: increased oxygenation of the blood flow, increased function of circulatory system. We can be faster, we can be more alert, we have higher oxygenation levels, except for birds compared to birds. And so we have in general a set of evolved body structures that are shared by most vertebrates and indeed most mammals, although we have these evolutionary differences that have allowed different kinds of mammals to adapt to different kinds of habitats. And also we have flexibility in our physiology so that we can adjust and maintain homeostasis, whatever the environment throws at us.